So good morning. So we'll we'll continue from what we left with yesterday. So what was the thing which we were covering the other day? It was all to do with the solid and liquid side stream and how we get different type of side stream from both seafood as well as macroalgae, and we did not cover much of microalgae, but your macroalgae, shells, seafood, and what are the side stream. And liquid stream was mainly related to processed water and wastewater and how we can valorize that. So we'll and there was also a practical demonstration for shells valorization. So what was practically done yesterday was shells, which colleagues from Rourkela, they got that shell, they washed it, they already dried it. What they did not do was the grinding part. Yeah, if you grind it, you reduce the particle size, dissolution becomes faster, and then this step was eliminated because if we employ ethanol, then you can get all those pigment. If you have seen the color, orange color or different type of color was there, and some pigments are colorless. So ethanol dissolves those pigments. So we did not do that. Yeah, because don't wanted you guys to handle with ethanol because all are not from the practical background. Yeah, and then what was done, you dissolved hydrochloric acid, and what other acid was used? Acetic acid. Yeah, so if you, how many of you have added the egg directly to vinegar? If you add whole egg into vinegar, leave it for 24 hours, you will see the shell is gone. Yeah, because shell contains calcium carbonate. Yeah, when it comes in contact with HCl, what happens? You would have seen some of the bubbles forming. Yeah, that is basically a CO2 which is escaping, and what is formed in a liquid is calcium chloride. Yeah, and if you have a food processing background, you know where the calcium chloride is used as a filtering aid, as a binder, another one. This happens with hydrochloric acid. If you use acetic acid, everything happens the same, and what you get is calcium acetate. Yeah, right, and whatever is left over then is your protein plus chitin, because that is a robust network of minerals, proteins and chitin. So every shell does not contain a chitin. Chitin, protein along with the calcium makes very hard shell. That's why crab is having harder shells, shrimp is harder, whereas other shells what you see, mussels and snails, they have very soft shell because they do not contain any chitin there. Yeah. So what happens after this? This step is called demineralization. Yeah. So now whatever shells you will be seeing, it will be a bit softer. It may be possible it is not completely demineralized because of number of different factors. One of the factor is the ratio of it and the concentration of acid. That has to be optimized based on each and every species. So what ratio you used was 1:20. Yeah. So if we use ratio of 1:50, it will dissolve much better. Can you guess why? Simple stuff is, if you have 1 kg of sugar, how long will it take to dissolve in 1 liter of water? Bit longer. Yeah. But if you add it into 50 liters of water, it will dissolve straight away. Yeah. So that is saturation level. That will be dependent on what is the saturation of end product into the liquid suspension. Yeah. So that is estimated either based on the calculation or based on trial and error method. So these are the things which are optimized each and every step. Yeah. So once you have this one done, you wash whatever residue is left over. Yeah. To remove the acid, then you add alkali. Yeah. The moment you add alkali, all the protein will be dissolved in alkaline solution. Whatever is left over is your chitin. Yeah. And this particular protein can be precipitated back with HCl to get a protein biomass coming out of shells, though it will be very small quantity. Majority of the composition of shell is calcium carbonate, yeah. And this chitin, if you mix it at very high temperature, it forms chitosan, and chitosan which is sold, yeah. So you have seen how the sequentially the processing is done, yeah. So maybe lecture 15 onwards you will see all the new technologies of usage of microwave and other processes, yeah. So that's the way it can be. Microwave, ultrasonics, high pressure, all those technology will help you to accelerate either this reaction or accelerate this particular reaction. Based on that, we deploy technology. Either we grind it. If you are grinding it, you are introducing energy into it. If you can use whole shell, it's much better, even though if it takes a bit longer. Grinding hard shell is very, very difficult, yeah. So we have done some of the innovative activities. When we have these shells, we sprinkle acid on it. As we dry it, the acid will weaken the shell. So the grinding energy is reduced, yeah, during the drying process. So these are all the interventions which can be done to improve this particular process. So this is your baseline process and how you improve your process based on technology, yeah. So if your monthly budget is 1000 rupees to spend, is up to you how you improve your quality of life. Either you spend everything in the first place, day one, or everything at the 30th of the month, yeah, or you have proper planning and distribute your funds so that your quality of life is different, yeah. So what we are going to cover today is what is duckweed, yeah. How many of you know about duckweed? It's not a weed which duck eats, but that's the name of the common name of a plant. Cultivation of duckweed and how we can use duckweed as a valorization tool for wastewater, yeah. So duckweed is actually a small aquatic plant, yeah, which is having a commercial cultivation mainly in US and Israel. We already discussed this. And you can get up to 30 to 40 tons per hectare and almost 55 tons per hectare, up to 55. So these are some of the statistics how you grow. And it can be grown on wastewater. What is microbiome? That is microbial, so in microbes in biosphere. Diversity, stability with significant impact on duckweed growth and nutritional composition. So all these duckweed, whatever is grown, they have some unique features, and for that any standard book or any standard aquatic plant book, you can look into it and you will find all those answers. One of the common species what I will be discussing further up is Lamina minor or Lamina gibba. So these are some of the species which are quite extensively used. So what are the best benefits of using duckweed? One, they grow rapidly. They do not compete with the crop or land because they do grow on water. They trap CO2 from air, lower energy consumption, that means you don't need any input fertilizer, easy to harvest and low cost biorefinery, yeah. So these are some of the rosy picture of duckweed. Now if you will see, how many of you have seen such plant growing in any natural water, yeah. If you look around your cities or any waterway or stagnant water, you will see this type of plant pops up onto the surface. That is only because there is extra amount of nitrogen is available, or fertilizer, leaching of the fertilizer from agriculture runoff, or wastewater going into the water and it grows there. Either you will see greenish water, what you will see, what you might have seen in, if you keep a water in a glass bottle and leave it out, you will see some green algae is formed. That is basically a micro-algae, yeah. And now how the micro-algae is used now for range of different production. Same way duckweed also grows. Think of the wheat crop which is grown for getting wheat flour, or rice is grown. So what that how the wheat and rice was grown 500 years or 1000 years or 10,000 years or a couple of thousand years ago. Yeah, they were all grown in a forest. People, they picked it. That is called selection. Those who have genetics or breeding program, they take those crops, they grow it in a mass, and then they do the mass production. Very similar approach is being now adopted for duckweed, microalgae, macro-algae, to see what are the beneficial compounds you can get, and then you start a mass production. Currently, two countries, as I mentioned, Israel and US, they are doing large scale commercial production of these duckweeds. Yeah. So, they all belong to family Laminaceae. There are 5 different groups, something like same as wheat, Triticum durum, Triticum dicoccum, Triticum monococcum. The different species are there, and there are not only 36, there are large number of species are there. Some they have a small bit of root, some they do not have a root. They are mainly a floating plant, they float on the surface. Yeah. So, I am not going to go into each of these details, but these are just basic details about duckweed. That means how much it doubles. Some doubles in 24 hours, some doubles in 3 days, some doubles in 4 hours. That means within 24 hours their biomass is doubled. They work on range of pH 5 to 9. That means you can grow them even in a wastewater of dairy plant, because their pH is lower, or in alkaline condition, or on a neutral pH. So, this is very similar what will be the condition for any other plant to grow. 6 to 33 degrees Celsius, that means that again depends upon what species we are growing. Yeah. So, these are the some of the factors which are again from different reference sources, and if you are interested in, I can provide you those paper, or I can provide you the comprehensive literature review on this one, which is already in a publication domain. So, what I will be focusing mainly on those things which you may not find it in journals or in articles on from real applications. So, these are all temperature is required, sunlight is required, nutrient and a pH. Yeah. So, temperature naturally available, sunshine naturally available, nutrients from the wastewater, pH again depending upon the wastewater, or you alter the pH to grow that microalgae or to grow duckweed. So, this is the way it looks like. Lamina minor, it is having small bit of root, Wolffia, Lamina gibba, Lamina porphyra, Lamina, there are a lot of different species. So, as you can see how we start growing in simple duckweed in a flask, yeah, how it is grown in a tank, yeah, and how it looks like. And how it is used. If you grow it in in normal water, it is still consumed in most of the places as a food entity. This species lamina or duckweed, they contains almost 90% water, they have only moisture in it, very less amount of solid. Since they have a vacuole, so that is why they float in float on surface of water. So, how you can produce it? Even if you go in natural water, pick that plant, put it in the water, and you will see slowly it starts growing. Yeah, it's something like you bring in any plant and start planting it, and then from there you grow multiple plants. There is no rocket science in cultivation of duckweed. Yeah. So, as you know, all the scientists, how we are all known to complicate things, then you start looking into growing in different types of water, grow it in a milk, grow in a spoiled milk, grow in clean water, then you will see which one is better. Yeah. Based on that, you develop your own process, you get something interesting, you write a report, you publish it, that is the way it goes. Yeah. Or else you look into the literature, what people have done, you repeat it, it's just the copycat. Yeah. So, that is the way how innovation works. So, that is all simple duckweed, how it starts in lab production. Now, these are some of the complicated or advanced way of growing duckweed. As you can see, this is the top portion of duckweed, normal tank, you have dissolved oxygen, oxygen reduction potential, all these measurements are carried out, And this particular study found if you do have the aeration in the tank, even though you aerate it when you do wastewater treatment, yeah, aeration is required. So, when you aerate it, their growth is much faster and their nutrient recovery from the wastewater is much higher compared to non-aerated one. So, the intention here and our objective in this whole of the lecture module is how we can use this duckweed to remove nutrients A or to reduce BOD and COD of water and use that particular wastewater to grow a new type of biomass. Yeah. So, as you can see from here, these are some of the tanks which are cultivated, and this is the open cultivation or a closed cultivation. That all depends upon which country you are growing in, is a high temperature or a low temperature, because the temperature requirement is different. So, it is very similar to what way you have a wastewater tanks. And then in our Israeli industrial partner who is growing duckweed, they have changed the color of this duckweed from green to red by using a specific nutrient which is absorbed by a plant and becomes red. The purpose of having that red duckweed is to use as an alternative meat so that you get the same color, protein and everything. Yeah. How it can be made red? If you buy a white rose or a white lily, if you put that lily into red ink, what happens? All the white petals become reddish color or bluish color depending upon the ink. That means dye is taken up. Yeah. And if that is having a betalain or if having a beetroot juice or any of those things, that pigment or anthocyanin pigment is taken up by these plants. Yeah. So, you can change the color. Then you dry it, you have same red color. That was a very, very simple innovation, but it is very interesting. Yeah. So, it was not that somebody discovered a rocket or somebody discovered a new equation how to launch a rocket. Yeah. So, that is the way you can see from the image how we can grow duckweed. So, these are again lot of new ways of doing studies and production are ongoing. So, as other day I was mentioning about how we are cultivating at a larger scale. These are all for demonstration site, as this is Mount Lucas in the center of the country, and here as you can see these are flowing water. This is not a small land, this is a couple of hectares of land. These are flowing water and they have a fish in different places. Yeah, fish is moving in this water and duckweed is grown on the top. All this is exactly what you see over here. Yeah, this duckweed is growing and also the energy is taken from wind, that is wind turbine. So, everything this is as a demonstration site to improve circularity. This is one of the place called peat land or also known as bog land. So, that is basically the soil is having so much of carbon, you take a soil and you can use it also as a fuel, because there is lot of embedded carbon. And as you can see, this is all duckweed and this is the man who is just collecting duckweed from the surface. Yeah. So, that means fish is also consuming duckweed as their feed and fish is also produced within that is trout. Yeah. So, you get both the things, how you can make a complete circle. So, as you can see, now this is what open land looks like. This is just a normal trawler which is just aerating it, and that can also be used for collection. As you can see, there are lot of other type of weeds are also grown. These are also aquatic plant. Then some of the plant operators, the people who are working there, then interest came in about this species. Let's see what is this species. Yeah. So, that is the way how things move. So, there is also issues with this particular protein. When you are growing in a water, there is also a possibility of shells growing in it. So, you may have a shellfish allergens in it. So, it again you have to be careful of all those nutritional labeling or allergen notification, whatever allergens it might have. And this is completely controlled cultivation. Yeah. And same cultivation can be in a moving water, how you treat it. Yeah. So, this is open pond system and this is closed. So, how difficult is to grow duckweed? Go to the river or pond, pick one plant, put it in the water, in 2-3 days it will double, then from there you start cultivating. From one seed, one wheat seed, how many seeds you can get? After 3-4 months, you can get more than 1000, right? And from 1000, you know how much you can get, right? The same thing works here. So, there is no seed for this one, but whereas a small inoculum or can be done with the spores and different way, like what when you see the seaweed cultivation, macro-algae cultivation, you will be able to find that. Where all we can use it and how we can grow this. Clean water is easy, yeah, but that can also grow in slurry, animal dung slurry, food processing wastewater, swine, because you know wherever swines, that's pig farming is, you have all those lagoon effluents are there, you can grow all those duckweeds there, yeah, or any of the wastewater. They all have a problem of high BOD and COD. You adjust the pH depending upon species you grow. Nutrient recovery, you may, if you are looking for a large amount of production, then you may have to see how you can adjust calcium, magnesium and all those ratio, and that can again complement from your the experiment what you are doing the other day, yeah. In some cases in agriculture, eggs, normal eggs or rotten eggs are buried into the soil. For what? So that calcium and magnesium, all the nutrients are embedded into the soil and crops can take those, yeah, rather than supplying external chemical fertilizer into the soil, yeah. And then this is the way duckweed cultivation can happen, and then you get a biomass which is rich in protein. It removes the nutrients from the wastewater. That means inorganic form of nitrogen, that is nitrates, nitrites into proteins, yeah. Carbon forms from CO2 from the air to make into starch, cellulose and the nutrients. So, that is what it is, yeah. Is it complicated to grow duckweed? It is very straightforward, right. So, if you will see how much duckweed can remove or can be produced, slaughterhouse wastewater, that is animal abattoir, 330 grams per square meter after 30 days. This is dry mass. Oil palm effluent, just depending upon what they have reported in different studies. Chemical oxygen demand for the wastewater reduced by 18%, CO2 85% after 28 days. Is just again depending upon what is the intention and how different types of water can reduce. So, this is the way you can use your own wastewater and cultivate it. And remember, this is all the agriculture wastewater. Think if you are involved in protein extraction, you have lot of wastewater or processed water generated after protein extraction. Like what you have done the experiment, you have acid solution. If you remove the calcium chloride either by membrane or even now you will be going to do drying part, that water you are going to dump it. That water is having some of the nutrients and that have a specific pH. You can select a strain to grow duckweed on it, and that is the way circle can be completed. So, that works on circular economy principle, that means closing the circles, yeah. So, that is one. Once production is there, harvesting, simple scoop, the way you take a cream from the milk surfaces, yeah. You do mechanical harvesting or you harvest it at a regular interval depending upon how much biomass is produced. So, it is a continuous process, that means wastewater keeps coming in, yeah. You keep recovering your duckweed. So, it is not that the wastewater is there, only certain amount of nutrients are removed, then you dump it back. So, it can be a continuous process. continuous supply of wastewater, continuous production of biomass and continuous harvesting. Did you get the purpose right here? Yeah. So, if you have a large area, so you are continuously getting biomass, whereas if you are using the wastewater treatment system, what you are getting continuously is the sludge which has to be disposed at some point, some way or the other. Then again, wherever you are disposing, then you have aluminum and all those chemicals which are going in. So, with this you are not adding any nutrient or you are not adding any input chemicals, because when you add any chemical, flocculating agent or so, there is also a cost and environmental impact of producing those chemical is also associated with life cycle assessment. Yeah. So, 6 to 7 grams dry mass per square meter per day. Yeah. So, it's a dry biomass, because wet one will be quite high. And what type of protein this contains normally? The protein in this one is mainly Rubisco. So, normal protein content is around 25 to 35% in this duckweed on dry basis, and half of that protein is Rubisco. So, you do not need to use any alkaline extraction or anything, simple squeezing of its juice, precipitating the protein, you get more than 50% protein in it. So, you can classify it as protein concentrates coming out of it. It also contains starch, it also contains fiber, it also contains different bioactives, and those bioactives, they do have antimicrobial properties, and there are some IPs which I cannot go and discuss over here. Some of the bioactives from this particular plant are also used as a biofertilizer for different agriculture crops. Yeah. Anything which grows in very high amount of water, they have very good antibacterial property, because they do grow in a water and bacteria needs higher amount of water to grow. So, that is whole open area of research to explore, yeah, and do something interesting, something new. Otherwise, doing the same bit of things what are already there in literature, already people are doing, something like keep chewing the chewing gum, even the sweetness is gone. Either you can't swallow it, can't spit it. Yeah. So, duckweed application, this is again is taken from, this has been taken from CGB bioenergy, duckweed, fermentation, your bioethanol, biogas or biohydrogen production, because it contains starch. Once you get a protein, left over is fiber and starch, that is again for ethanol production, can be easily used for fermentation. It contains starch, so there can be a number of different applications. All these applications are non-food application, just because when we are using a wastewater, you have to go through the regulation for food application. Yeah. So, you have a lot of non-food applications. Then you have flavonoids, a different type of compounds can be formed, that is again bioactive compounds. You have protein for both animal feed as a protein powder, and these are some of the brands which are sold as a water lentil. What is the lentil? Masoor. Yeah. What's the botanical name? Lens esculentus. Yeah. So, same way this is water lentil powder. Then again other chemicals for different applications. Yeah. So, when we see this, automatically we think, oh wow, this is very nice, I should work on this. Yeah. Then you go to the next presentation, you say, oh, macro-algae is nice, let's do this one, right. Remember one thing, other side of the river is always green. Yeah. So, whatever you do, just be concentrated on that one, and there will be always the hurdles comes in between. Clearing those hurdles is the best part of it. Yeah. Never to jump on a topic, because you are here listening to duckweed, tomorrow it is water hyacinth, you will move to water hyacinth, then you move on to seaweed, then you move on to this one, right. There is a saying in Hindi, what you call dhobi ka kutta ghar ka na ghat ka. That means neither you can complete any of the work properly. So, just whatever you are doing, just learn the approach. Yeah. Learning the approach is important. Yeah. Dressing like somebody is not copying. Buying exactly the same dress is a copy. Yeah. So, that's, remember, that's not to get so intrigued about it. But if your focus is only on production of bioethanol, and that time you can consider different crops. Yeah. If you focus in a valorization of wastewater, then you can consider different crops. It depends upon where you want to use all this knowledge. Now, coming back to composition, so as you can see, this is 36% protein, this is gram per kg. Always be very, very careful with the units. Units are mix and match here, say milligram per gram of protein, yeah? And only 3% fat. So, it is again having saturated, monounsaturated and polyunsaturated fatty acids, some of those minerals. Now, when you see the lead, aluminum, chromium, all these things are coming in from the water. So, that is why these plants are also known as phytoremediation. That means you capture those heavy metals from the water to clear the water, yeah. And then that second question, what you do with this biomass, yeah. If you are concentrating it, getting only the ashing, so you are concentrating the minerals, yeah. It's not like in nutrition they say if you have iron deficiency, that does not mean you just go and start eating the iron rod, right? There is a biological form and there is chemical form. Hydrochloric acid is also produced in human being, in human stomach, but that does not mean you start consuming the hydrochloric acid. There is a difference between biological origin as well as mineral acids, A. B, when you see arsenic, how many of you know arsenic is dangerous? We know arsenic is dangerous, yeah, heavy metal. But the problem is naturally occurring arsenic or chemical form of arsenic, yeah. We do not have a method to segregate that. So, that is natural occurring one is still okay compared to chemically processed. So, these are some of the differences what we have to understand. And as you can see, everything is compared against soya, because in Europe we are looking only alternative for soya and they have a quite similar or a better amino acid profile compared to soybean. And this particular data is again depending upon the species or mixture of species. This one is what was calculated or is estimated from our lab from that particular site, but that particular site is not having a single species. The Labna minor, Labna gibba, the multiple species are there as a whole biomass. Those who are working in alternative protein, you would have come across on different research about leaf protein, grass protein, yeah. Our research group is also working on a grass protein which is all in a garden grass, because we have plenty of grass. The massive amount of green grass is there around the year because it rains most of the time. It's having a grass, and to extract grass protein, a very similar principle applied for duckweed to extract a protein, because majority of the protein is rubisco and several enzymes, yeah. And rubisco is very high molecular weight protein. So, what should be the procedure for extracting proteins? How do we extract protein? Anybody?
So, alkaline extraction and isoelectric precipitation.
Okay. So, what makes us to use alkaline followed by isoelectric point or isoelectric precipitation? Why alkali, why not acid or water?
Sir, alkaline helps to maintain the particular pH.
Okay. Well, that's true, but one thing is, before we select a solvent, this I will be repeating again when we go into the technology. One thing what you have to do is, first you have to do the protein profiling, yeah. It is very similar. If you want to take this whole table out of this door, what you have to do? First measure the size of the door and measure the size of the table, then you have to take horizontal or a vertical, right? Very similar way, before starting extraction of protein alkaline, it's just because majority of the cases proteins are soluble in alkali. You have to do one thing called Osborne classification. How many of you have heard this word? Osborne classification. I think everybody whosoever has studied basic biochemistry would have studied this. Yeah? That is solubility of protein. Yeah? Protein is either soluble in water. Yeah? So, those who are making egg alternative protein, fish alternative protein, meat alternative protein, they have to know first about this Osborne classification. Otherwise, it will be just wild guesses, cook and look type research activity. Osborne means one, certain proteins are water soluble. For example, albumin, globulins are water soluble. Yeah? Some proteins are salt soluble, that is 0.5% or 0.2 molar or 0.5 molar, something like that, 0.2 to 1% salt soluble proteins are there. Yeah? And then if you increase the salt content, protein precipitates. That is called salting in, salting out. You take some amount of salt to dissolve a protein, increase the salt concentration, protein will precipitate, right? 2. Third, alcohol soluble protein. Have you heard of it? Yeah? 4th, alkali soluble protein. 5th, acid soluble protein. Now, in fish, acid soluble protein is your gelatin or collagen. Yeah? Majority of the protein is alkali soluble. Some is alcohol soluble, some is salt soluble, and some is water soluble. Just now I mentioned about duckweed protein, majority is Robisco. It is a water soluble protein. So, if you use an alkali, your yield won't be that great what you get it from the water. Yeah? So, without that, using just a common solvent will not solve your problem. Yeah? Okay. So, that is why if you do Osborne classification, then you know what type of protein is there in a biomass. Luckily, you do not have to do it because majority of work is already there in a literature. But I always advise any of the guys who within our group working on a protein extraction, they do this first, because this is the important exercise to get different types of protein. For example, you would have seen some of the patent as egg replacer proteins from mung bean, yeah, by using cross-linking with sulfur or some cross-linking along with it, because mung bean protein is a water soluble protein and it is just exactly, it solubilizes easily. Yeah. So, that is the way you can get all the different type of water soluble protein, fat soluble, water soluble protein, acid soluble, alkali soluble, depending upon what is your application. If you are making a beverage, if you want to add protein to orange juice, so you can only add that much amount of protein which is soluble at that particular pH. Yeah. So, basic background of any of those solution or solvent is very important before you start thinking, let me extract it by using ultrasound, microwave or anything. Yeah. So, majority of the protein is alkali soluble, that is why we think alkali is the best solvent. Yeah. Now, this is lamina minor fresh duckweed. When you freeze dry it, you get a perfect green powder. Yeah. If you sun dry it or you hot air dry it, still you get slightly darker color. Yeah. This is what normal most of the experiments are carried out. Then you have different types of technology by using normal stirring, what you do most of the time, if you add alkali or water to extract it, or use ultrasonic systems or use high pressure. I just giving a brief example how the protein is extracted from duckweed first, then starch or a fiber, or else if you take this is the normal protocol what you would have followed in your experiments if you are doing the experiment. What I would do differently, I will take this, I will get the juice out of it, use a very nice juicer to squeeze all the water. Yeah. When you squeeze all the water, what you will get? all the water soluble fraction, and you leave it for some time, you will see starch precipitates at the bottom. Yeah. How many of you know how to extract the starch? Again, very simple. If you peel a potato, you will see white color powder. What is that? That's the starch. Yeah. That means starch is embedded like a globules within a matrix. Yeah. Because that's a storage. It stores all the glucose in the form of starch. The moment we get a juice out of it, you leave the juice for certain time, all the starch will precipitate. Liquid portion, if you dry it, use a membrane, you get your protein. The starch and protein is gone, then whatever is left over protein and a fiber is now your alkali soluble protein, then you can use alkali extraction process to get the second round of protein. Yeah. That's the way a biorefinery processes are developed. Yeah. Remember the sequence, what should come first depending upon what is larger in quantity, or remove what might cause a problem with further extraction. That means you have to remove the lipid before you extract alkali protein. The reason is, if alkali adds with the lipid, it forms soap, then you will make soap. Yeah. So, we will come back to these technologies again. Yeah. These were all fancies of one of the PhD wanted to use all those technologies. Same thing like you all guys look into the paper. Oh wow, high pressure, let me try ultrasound, let me try this, and this is what you end up with. Yeah. But there could be a simple methods. You don't need a computer to get 10 plus 10. You don't need a calculator for that. Yeah. So, this is one of the example here. Now, yesterday and day before yesterday I was mentioning about Bioeconomy Student Challenge Award, which each and every country was sending one of those ones. So, we had a team of our one of the PhD student, 2 masters and 1 undergrad, they all went for this one. They kept its name as a phyto towel. Yeah. Whole logic was, dairy is a big industry in Ireland. Yeah. And huge amount of dairy is exported. Think in a country there was a milk quota. That means you cannot produce milk more than this quantity, because it was very difficult to use it or export it or to diffuse it. Yeah. So, large amount of milk goes for a cheese, and then they developed this type of phyto stacked system. So far whatever you have seen is single layer or a big tank, but you can do the same thing in a stacked tank basis. Mobile, modular, plug and play, scalable and fully automatic harvesting system. That means if water is moving, yeah. So, you can have some strainer, the smaller duckweed can pass through and larger will stay there. So, that is the way you can continuously harvest it and continuously reduce the BOD and COD in this particular chamber. I will quickly go through this so that it will give you a clear idea what I intend to say here. So, this is your all the light system, pipe, filtering conveyor, water catchment, biomass holding tank and pipe in, pipe out. And if you want to make it further more automated, you put a sensor here to measure its BOD and COD, and then again your machine learning, that means you fit in with an equation, yeah, to say that when BOD is less than 50, it should be discharged, otherwise it continues within the same circle. Yeah. So, simple idea from beaker how you can grow duckweed, you can make as complicated design as you want. Same thing, normal phone was 1G, then 2G, 3G, 4G. Yeah. What is the difference between 2G phone and 5G phone? And that is very important, because this topic will come in again. You know any difference between 1G, 2G, 3G, 4G, 5G or GIAN? Yeah? Do you know the difference? We all say, oh, I have 5G network. Do you know what a 5G means? Yeah? 1G was basically when you can only make a call. Yeah? First generation. 2G, phone and messaging. 3G, phone, message and internet. Yeah? That means all three can work together. In 2G you can also get an internet, but when you are on a call, internet disappears, yeah, right, you have noticed that one. 4G, all three will work together at a higher speed, and this was very, very high speed, yeah. So that was the only generation difference, because obviously when you are on a phone, you don't need an internet. So that's why all these generations were there, and you will see this reference of this one when we go through the protein extraction process, yeah. So this is what I have referred also as a protein, first generation protein, eating normal meat, pulses, legumes, that were first generation. Second generation, whey protein, all these, whey protein isolates, protein concentrates, second generation. Third generation, your insect proteins and all new sources of protein coming in. Fourth generation, how we are going in changing our protein in our diet. Earlier what was it? You eat a protein rich food, healthy. Now you need a protein supplement. Generations, first, second, third, fourth, fifth generation. That's why we classify as protein juggernaut, yeah. Now coming back to this, so this was a simple design. This is the repeat of the slide. That means either you go for conventional or go for simple circular system, yeah. So this image what you saw in 6th or 7th lecture, this was the history, as now we are going through, yeah. So sometimes I do take movies approach like a Bahubali, first and then the conclusion comes in, right, the same thing. So something happens and then you go back to the history, so that you won't forget it, yeah. So how this all came in, all the way from simple beaker, and for that you don't need to have a biology degree to understand how to grow a crop, yeah. Farmers, when they grow a crop, they do not study agriculture or agronomy, yeah. They learn by a practice, and then to find the answers to the questions. In search of that, you develop scientific principles, yeah. Because this could be a controversial statement, science is always reactive, yeah, not active. Things were happening, we draw theories based on that, and then we say based on this theory this happens, yeah. So what was happening? Apple was falling before the gravity was discovered, yeah, correct. It's not that when gravity was discovered then only apple started falling. So sometimes there is always an, oh, you believe in this and you are a scientist, yeah, right. So science is always reactive. My name is Brijesh because my parents calls me Brijesh. If they would have called me Tomato, I would have been known as Tomato Tiwari, right. So that's the way the processes and everything is developed. So anything, whatever happens, you find an answer to it, you develop an equation, and then you believe it is always going to be this. If you would have studied the first order kinetics, yeah, every growth is exponential growth, yeah, microbial growth or anything. It cannot happen if microorganism is growing, all of a sudden it dies and then again grows, yeah. So that means everything will go in first order. First order because it will never touch zero. How many of you have studied half-life? What is a half-life?
So reduce the radioactive by half, 90%.
Okay. By 50%,
yes,
yeah?
Sorry, yes, sir, sorry, sir, 50%.
So that means 100% will be double of that. Why?
Like in next cycle, it will again reduce 50% of it.
It will never go zero, right?
Yeah.
Why? Because it's first order.
Yeah.
It's very simple. It's nothing. I know your formula is 2.303 upon k, yeah. But think exponential equation is what? Population at any time t, yeah, is equal to initial population, yeah, exp minus kT. Yeah. Same thing, concentration at any time t is equal to or directly proportional to initial concentration exponential minus kT. Why minus? Because it is reducing, right. So, you said half life, that is Nt is equal to 1 by 2 or N0 by 2, right. If you put N0 by 2, N0 exp minus kT, yeah, gone, 1 by 2. Now, this 1 upon exp kT, that is why we use minus. Yeah. In maths, signs are used. So, remember, data is for maths, math is not for the data. Yeah. Thing happens, we fit an equation. If you fit a best equation, that is the way model works. Yeah. So, that means exp kT is equal to 2 is natural log. Yeah. ln of k, then if you take natural log both the side, this becomes kT, yeah, is equal to ln 2, correct, t half life ln 2 by k. ln 2 is what? Natural log of 2 is 0.693, simple. Yeah. And this is what we did study when we were 8th or 9th class, half life is this. Yeah. And we always remember this formula, amount of time required to reduce the population by 50%. Yeah. Next 50%, that means it will be like this, that means it will never go 0, that means nothing can be destroyed from this earth, back to the thermodynamics law, that means we use exponential one, and moreover log of this is not defined. That is the only reason why we use these type of numbers. And same thing happens with the, why I am touching this one again, because when I go through the technology section, my life will become bit easier. Yeah. And you may have some more questions coming in. So, how many of you have studied microbiology? What is the D value? In a mic here, please. Yeah.
Sir, the time taken to reduce 90% of microorganism at a definite temperature or constant temperature?
90% population. Yeah. That means 1 log cycle.
Yes, sir.
So, 2 log cycle will be?
Sir, 2 log cycle again, next one, sir.
Tell me how much?
Sir, next 90, I guess.
Yeah, that means if I have 100 microorganisms, 2 log will be how much? 90 or 91 or 99?
98.
It will be 1.
So, 100 becomes 1, yeah. 2 log means 99% removal, right. 3 log 99.9, 4 log 99.99, yeah. In the same way, food processing comes 12 log, yeah. That means 99 point so many nines, but still not 100%, yeah. Now, why we use log number in microorganisms? We say no log 5 log, yeah. We have only 2 log reduction. 2 log reduction means 99% of population is removed, but still if you take any of the food processing book, you need to have a 5 log reduction, 6 log reduction. Why? Pasteurization is 5 log, sterilization is 12 log, yeah. Why do we use log number? Why don't we just write it directly how much population is there? Any idea? Again, simple assignment, think over it. When we go into the technology for reduction, that time will go to that one, yeah. Why do we use log number for microbial population, yeah? Why not actual number? Population is 3, 7, 8, 9, 4, 6 bacteria, yeah, and we use unit called CFU, that is colony forming unit. in one bacteria can form one colony, one bacteria can form 1000 colonies. Yeah? Other way around. One colony can contain one bacteria and the same colony can contain 1000 bacteria. Something like Delhi is also a city, Rourkela is also a city. Yeah? Population is different, but city is something like CFU. Yeah? Come back with the argument, why do we use log number? I will make it further more complicated. One of the argument could be it's a large number, that is why we use log numbers, right? But have you ever seen your blood reports? 5.9 into 10 to the power 6 cells per ml. Why we do not use log number there? WBC, RBC, all these blood count and all other counts of human body, they are exact numbers but not log numbers. Think over it, yeah? And if you find it in any other journal, let me know which journal has written that information. That is all is there in basic books which we often ignore. So, coming back to this one, this was all the pitch, high protein for animal feed in duckweed, environmentally destructed animal feed if you are using soybean crops, because it is not really environmentally destructive, but at this stage it is, because we need more and more land to grow crops. Population is increasing, we do not have a land, so start cutting the forest. So, we are disturbing environment, whereas for this you do not need land. Yeah? So, whatever all this fertilizer, when you cultivate it, you have again environmental degradation, whereas all these type of phosphorus, calcium and everything is recovered from the wastewater and goes into duckweed which contains those minerals. So, that will supplement. Yeah. Now, first question, duckweed is easy to cultivate. Yeah. Second, processing is very simple, you just dry it and use it directly for the feed purpose. Or duckweed is grown, if you have a fish farm, same duckweed is consumed by the fish directly from there. So, you do not have to go and make a protein for fish feed. So, this is called circularity, that means to introduce the short circle. Yeah? You cannot achieve complete zero waste, but that is the way we can get it. Now, I will quickly go through again with the practical, some of you were not there present in the beginning. Yeah? So, basically what experiment was done yesterday was this part. Yeah? You had the shells. Yeah? Who is going to repeat it for me? I am already bored now. Who? Who was awake when I was doing this? Quick, quick, quick. No? You? Who want to repeat it what we did yesterday or you did yesterday? Okay, I will repeat it anyway. So, shells were washed and dried, what you got it, yeah? And we eliminated this particular step because do not wanted you guys to handle ethanol, and this would have taken away your astaxanthin and some of those pigments which were colored. Yeah? Alcohol can remove most of the pigments out of the biomass. If you use a methanol, even better, it can remove those pigment. Ethanol, methanol, diethyl ether and all those solvents, whatever you guys were telling me, all those solvents can be used to remove those pigments. Then we are adding organic chemicals, and subsequent food and non-food application becomes trickier. So, we eliminated this. So, you had your hydrochloric acid, mixed it, and duration can be overnight, 2 to 3 hours depending upon the concentration, solid liquid ratio, temperature usage, stirring, non-stirring, all those things will dictate how much time will be, that has to be optimized. And when we use hydrochloric acid, what is the composition of shell? calcium carbonate, chitin plus protein plus others. Yeah. The moment we add HCl, calcium, and this is broken down into CO2, goes into the air, and you get CaCl2. You can do the mass balance very, very easily. You know how much calcium is there in a form of calcium carbonate. Get all your chemistry books opened, simple analytical chemistry or physical chemistry. Calcium chloride is formed in liquid suspension. Leftover biomass is chitin and protein. That means you are at this stage, and today your instructors will show you to wash it, and then they will add alkali, and this alkali will solubilize the protein. So, protein is out in one phase, calcium is out in one phase, and then if you leftover biomass will be chitin, and this chitin can be converted into chitosan, or even if this chitin is added to the soil, it will act as a biofertilizer because this chitin contains nitrogen. Even after demineralizing it, whatever is left over here, protein plus chitin, is also a sort of biofertilizer because it protein contains nitrogen, that also acts as a fertilizer. Yeah, make sense? Or half demineralized frustration, that means you do not have to complete demineralize it half, so that you have a calcium, magnesium, everything added to the soil. Then it becomes easier to handle in agriculture field, or whole shells can be added directly to the crop. So, those are some of the ways these type of biomasses can be used. So, we will take a short break, and we will come back at 11 o'clock. Yeah. So, try to come in a time. So, what did we cover just now? Duckweed. Yeah, is easy to cultivate. Yeah, there is no rocket science that you don't have to get special species and other things to do it. Yeah. Now we'll go on to the production of macro and micro algae. Yeah, and how many of you have seen algae? You all, yeah? Which one, micro or macro? Now, first question itself is, so far whatever we were talking about, all the blue economy, blue circular economy, and blue bioresources, do you think is sea a sustainable solution? So far whatever we have covered, almost 9 lectures, you would have seen oceans are going to be the one going to solve all the problems. Yeah. But in theory, based on the history, it is because all the life originated from water. Yeah. That's the reality. So, the two important things when it comes for sea is the focus on biomass and how we maintain the biodiversity. That two things are very, very important. If we are doing overfishing, yeah, then we are eliminating certain nutrient from the sea water. So, that is why the balance between these two becomes very, very critical. Yeah. So, that is the reason why we have to have a proper balance, either you go with aquaculture or a wild harvest. So, when we do commercial harvesting of seaweeds, yeah, we already covered what is wild harvest and aquaculture. Those who are new, you can look into the previous one, I do not know, it should be available online to see what is difference between wild harvest and aquaculture. So, the problem with this wild harvesting is what are the waste produced from commercial seaweed harvesting, yeah, which is grown naturally. if you remove all those wild harvested seaweed, then there is a problem of again silting, and these seaweeds they bind the shores, otherwise lot of sand and everything will be migrating. You know all those issues of silting of rivers, removing of the sand from river bank causes flood and other things. There all purpose is there, naturally all those things builds up, yeah, and also prevents from storm. That means it protects us from storm and other one, right. So that's why it's very, very important when you do harvesting. It includes lot of different types of licensing. Now this is actually from County Kerry, yeah. It's very, the whole of Irish population is around 5.8 million, yeah. Out of that 75% of population lives in 3 cities, yeah. So all this area, if you will see, this is the sea and this is all seaweed which is there on different rocks, yeah. People they go in and they harvest. This man's name is Henry J. Loynes. He was a professor and dean in a university, and he started 4 or 5 different companies of seaweeds. And then it's a big business at the moment. He likes all this. So by looking at the seaweed, he can identify what type of seaweed it is, yeah. To some extent I can, yeah. Like if you see the rice, oh this is a basmati rice, this is not a basmati rice, this is Dehradun basmati, this is pony rice, you know. Farmers they know it. You ask scientists, they say, oh I need to test this aroma, that profile and all, yeah. So there is a difference. All those guys who are from dairy, what is the difference between cow and a buffalo? Don't tell me the color, don't tell me about the milk, yeah. If you see an animal, how do you know this is a cow, this is a buffalo? There is only one difference between cow and buffalo. What is that? Physically. Anyway, cow contains collar, buffalo doesn't have a collar, yeah, right. Horn is there, color is black, yeah. I know skin wise and all you can identify, but physically there is only one difference. But you say the milk is thicker compared to this, cow milk is yellowish color, right. Same way some of the identification of the seaweed is also there, like I mentioned some of the species previously, that is Escophyllum nodosum, yeah. This name came in because it contains node, Fucus vesiculosus, yeah. And there are different types of names are given, broader classification, green seaweed, brown seaweed, red seaweed based on their natural color, A. Another classification is given based on what is their structure like, bladderwrack, yeah. And then other ones are Alaria. So there are number of different species depending upon the geographical location they grow. And broadly they are also classified as kelp. Sugar kelp, Saccharina latacema, yeah. So that's sugar kelp. So kelp, seaweed, macro-algae, all these are the different names of the same biomass, yeah, right. So that's for, and these are mainly grown at underwater dense forests. You would have seen the video other day. So they start from the bottom. They can be as long as 2 meters and 3 meters big, long fronds. Fronds means like how it is there, and it is attached to a surface with hold fast. It is very difficult for you to remove that seaweed from the rock. They attach very nicely, like a snails. If they are attached to the rock, you can't remove it just like a normal hand. It is having very good adhesive power coupled with the compound it contains plus the vacuum it creates, yeah. So that's what builds with dynamics of ecosystems on earth. So wild harvest which grows and is just cultivated directly, that is harvested manually or by mechanical manner, and this is completely mechanized way of harvesting. So I'll go through this small bit with the production and then how we process it. Now what is really an economic value of seaweed, because this has been quite important. One, it can address the issues with protein deficit. food shortage, population is growing and the climate change. So, we need to produce crops which are carbon neutral or carbon negative. Anything we produce that is having a impact on a climate, whether it's rice, wheat, any other crop what we grow, they have impact on environment, because we need a land, we need a fertilizer, we need a resources. Whereas seaweed does not need that sort of input, because you just throw it into the sea, it grows on its own. Yeah. So, as you can see, these are again the statistics how the wild harvest remains stagnant and aquaculture production is increasing. So, we discussed about aquaculture as IMTA system, yeah, integrated multi-tropic aquaculture. That means you grow seaweed, you grow shells, you grow different fish all together, yeah. Whereas you can only grow seaweed also, and it solves lot of aspects related to United Nations Sustainability Developmental Goals. And as you can see, among number of countries, how much aquaculture is there in near about Asian subcontinent. China is number 1 in producing seaweed. Indonesia, Philippines, Korea, Japan. Korea, where is India? We do not have much. It is still there, but not huge. So, there is a huge potential again. We have long coastline and to produce aquaculture seaweed. Yeah, may not be for food purpose, for other application, alginates, carrageenan, all those biomasses or those ingredients can be obtained from seaweed. At the moment they are extracted, they are produced. There are couple of institutes here like in Kerala as well as in Gujarat, which deals with seaweed aquaculture as well. So, if you will see the economic importance, how much seaweed is produced and harvested. Yeah. So, first one is the key ingredients what you can get out of it: carrageenan, agar-agar, alginate. You know about agar-agar, which is used extensively as a microbiological media, but that is also used for food purposes. All those jellies, sugar jellies, they all contain this type of stuff. Same thing, alginate is also used for encapsulation of yeast for fermentation. That's sodium alginate is soluble in water, calcium alginate is not soluble in water. Food supplementation, minerals and dietary fiber. Yeah. How many of you have done experiment with alginates, creating alginate beads? Yeah.
Sir, in our laboratory, in plant tissue culture, we make a synthetic seed with the covering of sodium alginate.
Okay. Have you seen a brand on edible water? A lot of things were there with edible water. If you make 1% sodium alginate solution, a lot of applications are there for alginates, even for agar-agar, carrageenan in hand creams and everything. 1% sodium alginate solution, if you dip this water, you add anything to it. You make 1% alginate solution even in orange juice, apple juice or whatever juice you want to make, you make it. Then you also make 1% or 2% of CaCl2 solution. Yeah. And this was formed in shells. Yeah. The moment you drop sodium alginate beads into calcium chloride solution, it will form a bead, because calcium alginate is not soluble in water, whereas sodium alginate is. Yeah. So, now you can make those vacuoles having a liquid inside and it is fully encapsulated biomass. Yeah. So, you can make edible water, because 1%, that means rest is water. Yeah. That is how it can be used for encapsulation. Add 1%, and if you want to do experiment, we can arrange here, you can see it. Yeah. Then you introduce your juice or whatever you want to introduce it, you do it, and then the moment it goes into calcium chloride, you will see small bead formation. Same type what you see in the market, if you just go on internet, you will see the edible water. That same water balls are made with sodium alginate dipped into calcium chloride, and outer one becomes calcium alginate, and in the center there is all water. Yeah. And it cannot be broken just like that. So, you can just swallow it, and alginate is consumed as a food ingredient. Yeah. Protein and all those things, fertilizer, plastics again, because agar-agar, carrageenan, alginate can also be used for making biopolymers, alternative to plastics. Because carrageenan is extracted, you take a seaweed, boil it at 90 degree or 100 degree for 3 to 4 hours, whatever is extracted in a water, you cool it down, it forms a gel. Yeah? So, that is basically a carrageenan extraction process. If you remove a carrageenan, left over biomass will be fiber and a protein. Then you use your enzyme to break the fibers, you get your sugars, then you have your protein. You will see all those things coming in next few slides. So, major producers China, Indonesia, Philippines and Korea. Yeah? Then environmental benefit. It captures the CO2 and inorganic waste. That means any of the carbon polymer formed in a seaweed, where carbon dioxide comes from? Environment. We have CO2 as a major greenhouse gas. Yeah? If 1 kg of seaweed is produced, yeah, and it contains 60% as carbon, yeah, that means amount of CO2 it has already absorbed from the environment and produced carbon biomass. Yeah. Then you have your much more yield compared to land. There is plenty of sea, you are not disturbing anything. Then you do not need a fresh water to grow this biomass. Yeah. So, there is an error here, growth of this biomass. Yeah. Then ecosystems, coastal ecosystem services, you have seaweed cultivation, it can also save your erosion, that is again you are going into this, and carbon sequestering. Carbon sequestering, carbon offset, carbon tax, carbon miles, you would have heard all these words. Yeah? Carbon sequestering is absorbing atmospheric CO2 and converting into some form of biomass. Yeah? Now, I did ask this question before, or I did discuss previously. If suppose there are 2 trees, yeah, or 2 plants, plant A and plant B, which one will absorb more CO2? How do we establish that? Oh. Do you want to answer it? Yeah, go on.
It's depend on how much, just like glucose is produced, biomass is produced, which species, like species 1 or species 2.
True. So, you know the photosynthesis process,
Yes.
right? Yeah.
Carbon dioxide plus water is
6 CO2 plus 6
6
H2O,
It will be converted into glucose,
C6 H12 O6 plus
plus oxygen.
right? So, if you measure amount of 6 molecules, or just do all the calculations, 6 into 12, yeah, CO2 molecular weight is what? Carbon is 12, oxygen 16, 16 plus 16 32, sorry, 32, 44 units of carbon, yeah, into 6 times of this will produce this much amount of carbon in the form of glucose. Yeah. So, if you measure the biomass of that particular plant or a tree or a species growth over time, yeah, that will show how much carbon dioxide it will need from the environment. Yeah. So, that is the way you can calculate the carbon sequestering potential of any crop. Yeah. Easier answer is I will look into literature. Yeah. But every crop, every plant on this earth is not available on literature. Yeah. But the basic way is measure the carbon, nitrogen, oxygen, sulfur content of that biomass by using elemental analysis, and then all these things are coming from nitrate, nitrites, carbon dioxide and all those fertilizer. That way you can measure how much it will quench. Yeah. Right? Simple. So, all these methods are simple. So, you don't need LCMS, MS to do all those measurements. Yeah? This is simple calculation, basic chemistry. So, if you will see this this is the European scenario. It's only building now to produce aquaculture. There are a lot of policies are there, a lot of blue economy things are going on. This is Iceland, this is Ireland, Portugal, Spain, France, all these coastal countries are going ahead with cultivation of micro and macro. You can see both macro-algae and micro-algae. There are some landlocked countries, they are trying to produce seaweed on land either by using fresh water or by using sea water, some form or the other. Yeah. So, this is all the statistics. 13 species are used out of 600 native species. There are more than several species are there. Some are invasive species which are coming through the water because water bodies are attached to each other. Yeah. And license, that means 180 hectare of land has been having a license, and getting a license is quite complicated because sea is not only used for production of seaweeds, but also for other aquaculture purposes, recreational activities, kayaking, scuba diving and all those things. So, licenses has to be applied. It is not that you just go and start using sea to do production. Yeah. And I have no idea about licensing system and rules related to use of coastal water. Yeah. Farmers, fishermen, they do not need a license. I think they do need a license about within the border, they can go and do the fishing. Yeah. There is no area belongs to an individual farmer, but somebody want to use that one to produce seaweed or aquaculture, they may need a license or they may need a permission to do so. It is very simple. If you want to open a physical shop, you need to have a shop license or you need to have to buy a shop. Whereas, if you have a trolley system, you do not need that. Yeah, that is why we call it grey economy and unorganized economy or organized sector. Yeah, so that is one. Now, when it comes to current production, and I did made a reference of BMRS, that is a company in Cork, that is Bantry Marine Research Station, and if you want to see all the projects, and they are the one which work with us on all the bioprocessing side of it, bio-refinery production and other elements. Yeah. So, if you have any questions, you can go on to the website, you can see the email. If you want to ask information, you can. Yeah. So, Bantry Marine Research Station, land based cultivation and sea based cultivation, the both are there for macroalgae. Here you have aquaculture, and if you will see, this is the sea and this is the way harvesting is done by boat. Majority of the harvesting is manual and rest is mechanical. How it is grown, we will see in next couple of slides. These are all images and you can find that, right. So, as I mentioned about Bantry Bay, that is Bantry is south of Ireland and Cork, and they have 16 hectare of sea site for licensing or licensed site to grow aquacultured seaweed. Yeah. Any idea in 1 hectare how much of biomass can be produced? Several tons, we will see that. So, seaweed hatchery, that means all these strains, they are grown first in a lab. Yeah, all those spores and culture, you will see that live, and then once it is done, it is roped around a single string of rope, that you can see. This is the life cycle of it. This is the full grown seaweed, similar to mushroom, the sporophyte, sporophylls, and these spores are attached to a single thread, and that particular thread is tied on to the rope. You would have seen that video the other day when we were running. Yeah, it's tied onto the rope and is thrown into the sea. Yeah, I think if you remember vaguely the video what I was playing. So, if you will see sorting in the lab, and these are what are collected from seaweed. This is a particular seaweed which is grown wild, and from there you collect a spores, put it on to the string to create sporophyte, and what you see here, this is basically a rope. Here this is a very thin rope Yeah, this is very thin rope which is tied together. This is dipped into spores so that all the spores are attached to that particular rope. That rope is very thin, you will see in a minute. And that particular rope, can you see this? This is lines are simple ropes, and these ropes are tied onto a thicker rope. Yeah, that means you have big thick string, powerful string made up of jute or so, and then this thin string is tied onto that rope. Yeah, like a thread. This is your rope and you just tie up with the thread, and the whole of that thread bottle was dipped into the spores. Yeah, that means something like a microbial contamination, and then this rope is thrown into the seawater. Yeah, and these balls, what you will see, are so that they are only floating. Yeah, they do not go down. That is the reason these are balls in something like this 1.5 meter, and this is the string which is connected to this water balls, which is floating, and the string is there in the water. That is it. Yeah, spores onto thin string, that string attached to a bigger rope and dipped into the sea. Deploying seeding string onto long lines at sea. So, if you will see here, these are the thin ropes, what you can see it, okay, that is clear enough. Yeah, because this one is much clearer than that one. Yeah. So, all the strings on this, and this particular one is dipped into the solution to get all the spores depending on species you want to grow, then tied onto this particular rope, and this rope connected to these type of water balls. You have seen that those balls are there in if you go in water sports, especially in near Marina Beach and all those type of how many of you gone what's called water sports place in Chennai, ECR road, water boats and all, you have gone there just to mark it, not to go beyond that. So, those are floating balls thrown in the water and attached with the string so that it does not move, and this could be hectares and long, could be a 100-meter long string or could be 200-meter long string. Yeah, this one is 110-meter long string and attached to a concrete block. Yeah, otherwise because of flow of water it will move to different places. Yeah, it's something like you have a rope to be hanged in a water. Yeah, this is for floating purposes and this is for having the rope at a specific point. This is what is done in boats, and then after 4 to 6 weeks this happens, and these are real picture from the production site, picture taken on 13th of January, and this is another one 1.7 meter 8th of January and 23rd of January. This was done to see the seasonal variation, year to year variation, and this is what happens, and this is what it grows like. Yeah. So, one single spore can create such a long seaweed, and final harvest is after 5 months. That means in February, and normally it is done in November and December, these ropes are thrown into the water, you forget it, and after 5 months you collect that rope and remove the seaweed. Yeah, simple. So, which one look more interesting, duckweed or seaweed? Yeah. So, that is, and then land based free floating, that one what you saw was Saccharina latacema, from where what we get? We get alginates. Alaria esculenta also you have alginate and used directly for consumption. All species are not 2 meter, 3 meter long. Some are the smaller species like vulva, the green ones, Palmaria palmata, Porphyra, Codium, Chondrus crispus, for Carrageenan, Glarcia. See, I know all these names now just because keep seeing it and keep talking about it. It's something like Saccharina latacema. So, that is grown in Norway, and all these alginate production, commercial alginate productions are done from those. Now, vulva, it contains high amount of protein, 29% is good, Palmaria palmata, and all these seaweeds, and some, as you can see, different aquaculture farms. Yeah. If you go and put aquaculture seaweed production, you will see plenty of different design whenever you get a time, you can go to China and you can see how the seaweed is cultivated, how it is dried, and the same rope because in Ireland is a cold country, you can't get it dried in sun. The same rope is hanged near the sea, it dries. You collect it, mill it, you have milled seaweed. Yeah, but if you have to use microwave drying or any other hot air drying system, it adds energy. So seaweed production cost increases. Yeah, so that's the way it can be cultivated, either creating an artificial water bodies, or this is at sea, or on land. Yeah, does it make sense? So if you have large protein production plant to produce a plant protein, leftover solution will be a salt water. Yeah, can we use that salt water to grow? I am not good at using the pen. To grow seaweed, or that salt water to grow duckweed, or to grow microalgae, or any other species to produce biomass of biological origin. So we are not competing with the land, and we are trying to get something cultivated without using any other resources. Yeah. So this is all your land cultivation. Now coming back to IMTA system. Yeah, you had a clear video and photograph in the previous lecture. Now this details you can find it again. This is a bigger project. This is a Portuguese company, Alga Plus, who produces this biomass, and they are making lot of vegan fish type product, vegan product, and using it in different product formulation. So this is again the street names, so forget about these street names. So then you have reservoir, you have a fish, sedimentation, and this is the way it looks like, how it is calculated. Sea bream and sea bass is a species of a fish, and it also requires whatever feed, and that seaweed or microalgae what is cultivated can be used for making fish feed. So what you are seeing here is how we can use different IMTA system only for seaweed. And again there is also an organic production of seaweed, because we are not adding any input, and there is an organic certification. So now market is shifting more towards organic ingredient, and if you have cleaner process to obtain carrageenan or alginate, so you have organic alginate, which is not there at the market, and soon you will see some of the products coming in the market. Yeah. So in that case you have to have an extraction process which does not include any of the chemical for extraction. Yeah. Organic potato is there, but organic starch is not there, because you use a chemical. But if you get a starch without using a chemical, you have organic starch, so you are creating a niche market for getting a premium price for that particular stuff. Now that was the rosy part. Alaria esculenta looks like this. This could be up to 1 to 2 meters long. Yeah. Laminaria digitata, laminaria, a lot of different species of laminarian is there. Saccharina latacema, it is known as sugar kelp. These are all abbreviation of the countries where these are cultivated. Yeah. Now I will quickly go through all the legislative issues. 1. Common fisheries policy. Alien species, that means you cannot take any species from one country and put it into other water. Yeah. If you would have seen in agriculture, lot of biomass we have in a country, weeds which are not origin to India. They just came in with the people, either they imported it or it came by chance. Yeah. One is I think cynodon dactylon, that is also not native to this place, I believe. Yeah. Certain seaweeds are Parthenium, I think. Yes, Pythenium is the one which is not of native, and it just came in by chance or by some seed or crop. So this is what it known as alien species regulation, common fisheries policy. That means setting out rules for management of fishing fleet which ensures conservation of fish stock. That means whatever fishing you are doing, you have to keep the stock on land, you cannot dump it back to the sea. Yeah, that means if you harvested the fish, certain amount of fish you don't want because they are dead or whatever, you cannot throw back to the sea. That was the one of the reason common fisheries policy was adopted. That's we call it as bycatches. Yeah, yeah. Alien species, again these are different regulation, monitoring of metals and iodine in seaweed. Now this is very peculiar thing. There is a iodine deficiency in India in northern part of the country, where salt is iodized. Yeah, whereas in southern part there is no iodine deficiency. Now iodine is a big problem in this seaweed when it is fed to animals or humans, because they get huge amount of iodine. Whereas if this seaweed is introduced into a human diet in the country where are not having vegetarian diet and they do not consume seafood, that can be best source of iodine. This is the place where I went wrong. I said, wow, this contains an iodine, we can extract iodine from here rather than using chemical iodine. But this is a regulation that it should not contain much of iodine. Yeah, it's a problem at one place and solution for other. What disease do we get if we do not have enough of iodine? Very good, and that's thyroxine. Yeah, so that is one thing to do, metal one, because this also traps all the minerals and heavy metal from the water. So you have to watch out what are the metals and non-metals. And if you want to see the application of seaweed for food and feed application, there is one FAO document which was prepared 2 years ago, and that included people from all over the world from different country. I was a part of this particular organization to prepare that particular document. It's freely available. Use of seaweed for food and non-food application or food safety aspect of it. It's a comprehensive document. It talks about the global level, not only for European perspective. Then you have maritime policy, framework, water directive, different regulations and novel food regulation. Yeah, and this is all related to Europe. Seaweed was used for cultivation for food purposes, then it doesn't come into novel food regulation. But if you are extracting an alginate or the species which is not consumed as a common food, will come under novel food regulation. Same novel food regulation also regulates the use of technology for production of those ingredients, and this is all governed by European Commission, or EFSA is advisory body, European Food Safety Authority. What is the body in India? FSSAI. FASI. What is the full form? FSSAI, sorry, this is wrongly written. Yeah. So they also have, FSSAI is also having a very comprehensive document. It says what should be the regulation and regulates each and every entity. Yeah. So that's the benchmark for any businesses done in the country. But if that ingredient produced in country has to go to the Europe, it has to comply with this regulation, or if it has to go to America, it has to comply with this regulation. Yeah. So each country regulations are different. I'll give you an example of egg. Eggs are kept in a refrigerator in US, whereas in Europe eggs are not kept in refrigerator, because egg washing is allowed in the US, egg washing is not allowed in the Europe. Yeah. If you wash egg, whatever salmonella is there on the surface, it will enter in. Yeah, correct. So their regulations are different. So is the preservation strategy. In meat, you can use chemicals for preservation, whereas in Europe only lactic acid and steam is allowed for preservation, or no chemical. Fish, no preservative is allowed for preservation. Yeah. And now the decisions are going to come in shortly or maybe in a couple of years to eliminate chlorine altogether. A. Second thing which might come shortly is the removal of all the Teflon coating, you know, all the non-stick coatings is going to be banned because that contains fluorocarbons. So now alternative has to be there. How do you make it non-stick? Don't think about your pan for making food product. Think of the bigger baking ovens, bigger conveyor belt. How do we get alternative to this chemical? So there is always some problem to keep all the scientists in business. Yeah. So that's one, and the last regulation related to this is Habitat Directive, which is conservation of natural habitat, wild flora and fauna. Yeah. So that means, so these are the regulations all related to sea and all. They are not only these, there are multiple other regulations which has to be fulfilled. Yeah. Same thing, if you are producing, want to use the sea to grow seaweed, then you need to have all those licenses. For small amount of harvesting for your experiment, even some experiments were done here in NIT Rourkela, where we just harvested from the beach. But if you are harvesting tons, then you may need a license or need a permission. Yeah. 1 kg of sand from the sea you can bring it, but if you take a big damper or a big lorry, then it's a problem, right? The same thing applies over here. Any questions so far for macro? The only difference between macro and micro is what? Macro is done at a macro scale, micro is done at a micro scale. Is that correct? Yeah? No, it's not correct. Yeah. So just say it's not correct. Yeah. So any questions so far with macro-algae cultivation or duckweed cultivation? No? Good. So we'll ask a question, so each one of you will answer. Yeah. And I know who are saying no questions, so I'll ask them. Yeah. Now we'll go to micro-algae cultivation. Yeah. Micro-algae cultivation is as simple as what you have seen the duckweed cultivation. Yeah. So what you need again? CO2, light and water. It could be a wastewater, it could be a clean water. Yeah. And then you can get all this micro-algae, micro-algae for range of different applications. Yeah. So that is input, no nutrients required, you can add in nutrient, you have conversion system, all the technologies to produce a biomass, and biomass for either for food application, fertilizer or a feed or biofuel. There could be much more description of this. Yeah. Now this is too wordy. Yeah. And I'll be honest, this was these couple of slides were part of one of our own PhD who is producing microalgae from brewery wastewater. So we have a couple of PhDs and postdoc working on brewery wastewater to grow microalgae, to use fish processing wastewater to grow microalgae in combination along with a company which is based in Spain. Name is NeoAlgae, ALGAE .es, yeah, NeoAlgae, and all this thing is in impress-he.eu. Yeah. I already mentioned about this particular bigger project, and their octopus cooking water, well cooking water or seafood processing water, we are using either to grow microalgae, macroalgae or duckweed. Yeah. Now coming back to this one, nutraceuticals, which is very fancy topic, some of you are doing PhD related to that. Pharmaceutical, which is obviously to get pharmaceutical drugs, therapeutic protein and cosmeceuticals. Yeah. Therapeutic protein, that means they have lot of activity, certain protein has shown effectivity against viruses, and these are again certain extract which are inhibitors of certain enzyme which are responsible for your weight management or for metabolism activities and all those things. So, there are lot of activities are ongoing at the moment for microalgal extract. Same thing cosmeceuticals, moisturizer and all those hand creams. They do have lot of them. If you read through any of those expensive hand creams, if you read through, it will say seaweed extract or microalgae extract. Yeah, they do contain those in an ingredient form. You have antioxidants, skin whitening, fucoxanthin. Fucoxanthin is colored compound, microalgal extract. Phycocyanin is a colored protein. Vitamin, minerals and lot of carotenoids, and then you have different types of lipids. That all depends upon type of species. All seaweed will not contain everything, but depending upon the species. If you want to have, you all would have heard about spirulina, and you would have seen the cultivation of spirulina also. Water species, you collect it, you dry it, you have spirulina. It itself contains 60 to 70% protein, but still it will contain certain bioactives. You can prepare peptides and other application. Cholera vulgaris, cholera sorkina, then you have Purium perfidarium. A lot of different species are there. Each species they have different requirement for growth. So, that means depending upon your wastewater you can select that particular species. Oops. So, what are the first thing to do for cultivation of microalgae? You have to select different types of strains. Yeah, that depends upon what strain to select based on what is your target, either to remove the nutrient from the wastewater or your target is on a specific compound. So, if you want to have to recover the nutrient from the wastewater, you need to pick a species which is having very high growth rate based on the pH and the composition of wastewater. That is one. Second is on the product. What type of product you want? You want a protein focus, lipid focus or certain bioactive focus. Obviously, it does not need land for cultivation. You can do it in tubes, in circulating tubes, and you need a conducive environment. Same condition, whatever is required for growth of microorganisms, similar condition required for growth of duckweed. So, and all these things, when you get a strain, and there are multiple number of strains, whether it is genetically modified, naturally selected or selected in somewhere. So, one of the best ways, if you will see some farm where the wastewater is going in and you see those species are growing, you pick those species and try to nurture and cultivate. Yeah, that is the way, could be the easier part. So, what is overall process? One is open pond system, similar to aquaculture or duckweed. Photobioreactors. Photobioreactor looks a complicated word, but basically it is a reactor tube having a solution. You have something to purge air in it if it needs air. Sorry. If a species needs CO2, you purge CO2 and needs light. Photoautotropic, heterotropic, mixotropic, photoheterotropic. That is again depending upon the species. You can, some of you know this word, I am not going to go each one of them, based on that what is requirement for them to grow. Second one is how do you harvest it. Simple harvesting is, if you centrifuge it, cells will be at the bottom. Yeah, A. B, you can have a coagulation of flocculation, that means separate the biomass. Centrifugation, if they float, you collect it from the top or you do the filtration, because these cells are something in microns. Yeah. So, if you use microfiltration, you can get all those shells. Once you get those cells, that particular water can be reused or can be is normally clarified, or BOD and COD is automatically reduced. Yeah, that is it. And if you are focusing on extraction, either you mill it or use number of different technologies, which we will cover in coming lectures. Different technologies can be used for extraction purposes, or you convert these biomasses by using the biochemical process, chemical processes, or thermochemical processes. So, rest all extraction processing remains the same for whatever biomass you have. Yeah. Cell disruption, that is breaking the cells to extract a lipid, use supercritical fluid extraction to get the lipid, use a solvent to extract a lipid, or you just get a lipid for whatever purpose, food purpose or for fuel purpose. Yeah. Any question for microalgae cultivation so far? It looks like such a clean slide, I am just making it dirty. Yeah. So, so there are number of different ways seaweeds are microalgae is cultivated. Yeah. Race away, that means how the water is moving, their baffles, all these designs are based on to improve the efficacy. You may come up with much better design. Open pond, which is like a wastewater treatment system, or you have unstirred pond, stirred pond, you have plate reactor, you have column reactor. Column reactor, that means you have a funnel, measuring cylinder, that is also column reactor. Yeah. But when you call it a column reactor, looks fancier. If you say, oh, I did it in a beaker, yeah. So, that is all, do not worry about these terminologies. And if you have a light around it, you have photo bioreactor. Bio, because you have biological origin substance in it. Yeah. Technically, it is a simple column, or annular column, that means again if you have some of the beads into it, there are lot of systems are available commercially. If you browse through microalgae cultivation system, you can see hundreds of companies, hundreds of reactor. Technically, majority of them are just the configuration of different tubing system. Even if you will see the tubular reactor, yeah. So, the reactor, what sometimes I have seen is like a heat exchanger. Yeah, your heat exchanger, and water is I could have made better one. Yeah. And then you have a water going through this, and you have microalgae growing in here. Yeah, and the water is pumped back here with the new water going in, and you have a filtration system here to collect all the biomass. Yeah. And then you can have a batch type water going in. So, you can make it in this building all around with the tubing, and that is the way cultivation can be carried out. Something like hydroponics or aquaponics system as well. Yeah. Similar type of arrangement can also be done for duckweed. Now, here all these designs depends upon your size, your intention, and your timing and your purpose. If you are growing it for a very specific drugs or pharmaceutical bioactive or a protein, then if you are using your normal water for food purpose or for feed purpose. If you are using a wastewater, then purpose is completely different to produce a biomass. Yeah. Now, these are some of the advantages and disadvantages of harvesting method. Sedimentation, cells left to settle on its own, because when they grow big, they settle at the bottom. Low capital, do not damage cells, does not require chemicals. And then disadvantages. Sedimentation. Sedimentation compared to centrifugation. You do in a chemistry, what is the difference between when you centrifuge it or when you leave it on its own to settle? Mix a sand and a water, you leave it for an hour, it will settle at the bottom, right? You centrifuge it, you get it in 2 minutes. So, what will be the advantage of both the method? Same thing is here. Yeah, centrifugal force fast, this is slow. Yeah. So, there is no difference between normal centrifugation, sedimentation compared to this one. Yeah. All species chemical free and all this. Coagulation, flocculation, Same as wastewater treatment, how you create a flocculant, that means you aggregate all the cells together to make a bigger flock and remove it. Easy, fast, no contamination, it's physical method. Or contamination, large amount of area can cause cell death. Now, if the cell is only for recovery, not for further cultivation, you can use this particular method, flotation or filtration. Yeah. So, these are different types of harvesting method. I haven't gone into the details of it, but it's water, you have cells in it, and how you separate those cells from the water. Yeah. In lab, when you are proving the point, what is your microalgae and what for it can be grown, what all it contains, you will restrict yourself only with centrifuge, because every lab contains a centrifuge. When you go for scale up operations, then you have to think of a better way of doing it, or to use 3-phase centrifugal pumps, which is also used to separate milk, cream and of different fat. Like dairy, we use centrifugation pump. Same type of things can be used for this purpose as well. Yeah. Any questions so far?
Sir, you have told that duckweed can absorb cadmium, lead and arsenic. So, if we are using that duckweed to absorb those heavy metals, then what are we going to do with that duckweed? We cannot use that as animal feed or consumption. Again, it is going to be a matter can either be created or destroyed. Again, there are going to be a heavy metal contamination. Right?
Correct. So, any of the product, wherever any operations are carried out, there will be heavy metal contamination is there in every food product. Yeah. That has to be regulated with this, regulated based on how much we can consume. Yeah. Now, the question here is, if you see duckweed, microalgae and all, if your purpose of that water is goes into the sea, all those heavy metal, whatever is there in the water can be absorbed by the plant. If same element, same water goes into the sea, same water minerals will be absorbed by the fish. Yeah. So, that is why we also use these type of biomasses, not only your duckweed, even seaweed or even microalgae can absorb those heavy metals. That is why we also use them as phyto remediation. Yeah. That means how we can remove the heavy metal from the soil. Is there any other technique to remove heavy metal from the soil or from water? Yeah.
Sir, like in wastewater treatment, we can use activated carbon filters. That will be quick.
Yeah. Activated carbon, again the whole purpose here is whatever heavy metal is accumulated into duckweed, yeah, and then if duckweed is used as a feed purpose, and then if you are extracting the protein, that extracted protein may not have that heavy metal. So, everything has to be checked based on regulation. Yeah. Even if you are using activated carbon, yeah, if activated carbon is collecting heavy metal, after some time that activated carbon is saturated with heavy metal, then what we are going to do with that? Yeah. You understand my point here? Yeah. It is very similar principle applies how we can remove from the water. One is a reverse osmosis, remove all the minerals or so. There are multiple techniques are there. If your water is having heavy metal in it, that means that water used is coming in from the processing plant. That means your biomass itself, but suppose you have a dairy plant, yeah, any heavy metal coming into the wastewater, that means it is coming from milk, right. So, if this does not contain, then this duckweed will not have that heavy metal.
But in case of seafood, sir, there is a higher possibility of heavy metal contaminants
Yeah.
from the sea source.
Yes.
In case of dairy, we don't need to check for the heavy metal contamination, the chances are very low. But in case of seafoods, the chances are very high actually.
True, because seafood is actually, we discussed the word called biomagnification, yeah, you know, all the fuels and everything leaking into the sea, you have all the metal contamination consumed by fish, yeah, and then fish is consumed by humans. So that's why all those you would have seen lot of news report, the seafood from A, B and C country was banned because it contains huge amount of dioxins or huge amount of heavy metals, yeah, correct. So that's, these are still the challenges which need to be addressed, and duckweed or microalgae or seaweed are one of the way of removing those heavy metals from the water, because the time will come when the sea is full of heavy metals if you are using for transportation purposes. Same problem is also with iodine, huge amount of iodine is there, but if you are using seaweed for animal feed, and then the dairy product contains large amount of iodine. So this is the problem. Not only we are only talking about heavy metals here, but what about antibiotics as well, yeah. Yes. So that's why we need to have all those solutions and possibilities what to grow which can recover those heavy metals. There could be other way to concentrate the nutrients. If you have silver in a water and a duckweed is absorbing a seaweed, then you get your silver harvesting, yeah, you know. So that's could be on other way around, and that's very valid question, and you think and come up with the solution how we remove the heavy metal from there. And if that heavy metal, if you do see the ash, you know, total ashing how it is done, and then if that particular one, if you are extracting a protein in water, whether that particular heavy metal is attached to a fiber or it is attached to a protein, they are not freely available, they are bound in some form or the other. So there could be a way of removing those, yeah. If you consume a whole biomass, A. B, if a fresh duckweed contains 1 milligram per kg of cadmium, for example, which contains 90% moisture, if I dry it, this will increase, yeah. But you are not going to consume one full dried powder of a protein. So that's why regulations are set based on the proprietary product. Spices may have high level of heavy metals or or even salt will contain, like rock salt and all, which is coming from the sea salt or other one, they may contain all those heavy metals, but amount of salt consumed is minuscule quantity. So all those regulations are based on heavy metal per day per kg body weight, yeah. So it fits in the regulation because body can also digest that. Even we get lot of heavy metals in our body, you know, suppose working in the fuel, if you are walking in the water, lot of metals are absorbed through your body, yeah. Most of the waterborne diseases are not because of drinking water, but also working in dirty water. We would have seen that how the tapeworm enters into human body. So all those things are there, and that's, it's very valid question. So whenever we do any process or any processing, we know what is going in and what is coming out, yeah. Any other question?
Sir, is there any toxicity studies carried out for duckweed and microalgae from the biomass production?
Yes, there are plenty of cytotoxicity. Cytotoxicity, that means how much of this particular extract is required for any given function. If they have antimicrobial properties, cytotoxicity is done, toxic against the cell. All the anti-cancerous activity, yeah. If you have this as a cell, yeah, the moment you add the extract, yeah, this cell divides into two. So will it be cytotoxicity? It multiplies the cell, yeah. So it is not having anti-cancerous property, then this is considered as your wound healing property if it is multiplying the cell, because cancer is uncontrolled growth of cells, yeah. So if it is breaking the cells, it becomes cancerous property. So all the cytotoxicity that is what is the minimum concentration required to get antimicrobial effect and how much it will affect the human cells. So, any of the ingredient proposed either for human application, pharmaceuticals or nutraceuticals, cytotoxicity is must, yeah, and all the cytotoxicity on a specific ingredient, whether it is astaxanthin from seafood side stream, carotenoids, Purium perfidarium or Cholera vulgaris, all these extracts are done. And cytotoxicity is not only because the extracted ingredient, but also by the technique you have extracted. So, that is nowadays a must if you are going for any of those application. Yeah. You suppose take phycocyanin. Phycocyanin, that is color bearing compound, is orange color, purple color or green, all those things are some are the phycocyanin, some are chlorophyll. So, phycocyanin, if this is extracted by using microwave or ultrasonics or by plasma, it may have certain oxidation into it. Yeah. So, this is not because of phycocyanin, but because of extraction technology. So, cytotoxicity and their functional and biological properties are carried out once you are trying to prove the application of it. Yeah. So, even that is very important for any of the ingredient, otherwise there is no point in producing an ingredient which cannot be used for application, and literature is full of those cytotoxicity, and if you want we can go through with all those things, but it becomes slightly complicated. The reason I was saying here about anti-cancerous activity or wound healing, that is the basic test done to say if you put an extract, what is the dose required to break the cells, whether the or take for example 1 milligram per ml of solution is sufficient enough to cause antimicrobial activity. Yeah. And then you do the cytotoxicity, and what is the concentration will cause cytotoxicity. That means, if 2 milligram is per liter is a limit for cytotoxicity, then this can be used for as an antimicrobial activity or antibacterial in food purposes. But if cytotoxicity is 0.5, then this is not the suitable one. Yeah. So, cytotoxicity is a must to must be done in order to show the product application. Yeah. Any other?
Sir, what about the heat sensitivity of the duckweeds and microalgae, sir? Because you ask only the non-thermal processing for extraction of microalgae. What about the thermal treatment? Like the cultivation process and growing process is very easy and conventional method.
Yeah.
If I want to grow the microalgae in our pond, another thing, I go to the expensive method to extract that microalgae. So, any what about the heat sensitivity of that?
Okay. Now, heat sensitivity depends again on the ingredients. Yeah. If if you see the duckweed, for example, rubesco is very temperature sensitive. Yeah. And certain ingredients, when we are using the high temperature, they will lose their bioactivity. But certain ingredients are only extracted at higher temperature. Carrageenan, for example, you cannot extract it at the cold temperature. When we are saying non-thermal technology, there is a difference. We can classify it as a novel technologies. Yeah. For extraction, what is the most important principle? If you have a biomass and there is something trapped inside, in order to extract it, what you do? You add it into a solvent. Yeah. What is required? Solvent slowly goes into it, solubilize the target compound, comes into the suspension. Yeah. You centrifuge it, residue is out, what you will see when you go back to the lab. Yeah. And this is the extract. Now, you have to you collect it from this extract. When you use ultrasonic, for example, what it helps in allowing the solvent to get into the biomass. Yeah. It is not only for the temperature side of it, but also to improve the diffusion. If you have dry powder, before you extract, first thing is powder should be hydrated. Yeah. If you cook chickpea or anything, what you do? You put in hot water to soak it, what will happen? Outer surface absorbs the water, it doesn't penetrate in. Majority of the process, what is there? Overnight soaking, because slowly it penetrates into it. Yeah. Same way, if you put the same water in a microwave, the soaking same amount of water is absorbed in 10 minutes. Yeah. So the microwave assisted. So what you can achieve in 16 hours, you can achieve it in 10 minutes. That the purpose of the technology is where you want to heat or non-heat is second question. Yeah. One is you use a technology based on what is your objective there. Yeah. If suppose you are talking about heat, but certain antioxidant or bioactive compound, they are antioxidant, that means they can oxidize easily. If you are doing 16 hours of extractions, you are also adding lot of air into it. Yeah. So property is already gone or reduced to a certain extent. If you use ultrasonic or microwave, even though there is a thermal degradation, the thermal degradation is suppose 2%, yeah, but you preserve its oxidation properties. Yeah. So that is the balance what you have to do. Think of your thermal processing. If you can achieve 5 log, yeah, this is the curve for 90 degrees Celsius. Yeah. This is your inactivation curve for 70 degrees Celsius, and this is your curve for 110 degrees Celsius. Yeah. To achieve this much reduction, you get at 3 times. Yeah. A, B and C. Take it as 2 minutes, 5 minutes and 10 minutes. Which one you will pick?
2 minutes.
2 minutes, right? At 110 degrees. Why? It's shorter time.
Yes.
But this 100 and now it becomes a complicated one. This is take what product you want to take it as juice. Yeah. Now you do the ascorbic acid kinetics. Yeah. In 2 minutes at 100 and okay. Ascorbic acid 110 is like this, 70 degrees is like this, and 90 degrees like this. So that means 90% of ascorbic acid is gone in 2 minutes, whereas when doing for 10 minutes only 15% is gone. So selection of the condition depends upon what are the other things. If you read Louis Pasteur, the first book, I can make a product which is safe for consumption for 25 years, but there is no nutrients left in it. Yeah. So there is a compromise between economy and nutritional properties. Yeah. So that's the way when you select a process. So multiple unit operations are there. If you see milk powder, how many of you have made a tea out of milk powder? If you add a milk powder in a boiling water, what happens? Does it dissolve? For dissolving milk powder, you need lukewarm water, 40 to 42 degrees. Yeah. Food safety people want you to have it at 72 degrees Celsius. But to have a better solubility, powder properties, to have a flowability and nutritional and everything, you need lower temperature. Yeah. So what is the best solution for that? Which one you will pick? 72 degree, you can't make a tea out of milk powder. Yeah. So that's why you have to think. So what is the best solution for that? You can get rid of microorganisms also, and you can have a best mixing. You start boiling the kettle. This is what I do all the time when I have to make tea here. Yeah. And take whenever lukewarm water, a small quantity, make a milk. Yeah. When water is boiled, then add to it. Yeah. So the temperature overall itself will be 82. Once it is solubilized, the powder is hydrated, then you can increase the temperature and you will have the perfect milk. So same thing applies for extraction. Half the time when we say conventional extraction takes 16 hours compared to ultrasound 10 minutes, because in conventional method first 5 to 6 hours only goes in hydration of the powder. So there again freeze dried powder hydrates faster, hot air dried takes longer. If drying is done at 40 degrees Celsius, lesser nutrients are lost. When you dry it at 100 degrees Celsius, which takes less time, half nutrients are gone and it becomes harder. Yeah. So it's like a mess like this, yeah, okay. So all these things you have to consider before drawing the process. So one is unit operation, yeah, and one is to make a process. Raw material, washing, drying, extraction, yeah, spray drying to make a powder, yeah. Raw material like orange, you wash it, remove the moisture, peeling, and you just extract the juicer, then you spray dry it, you get a juice powder, yeah, right. But if the moment you open an orange, you get whole room smells like an orange. But if you open orange powder, you don't get that smell. So all those volatile gone in different stages, yeah. So when you make a process, you have to consider all those unit operations, then only it can be integrated into a different steps. As your colleague was asking the question about cytotoxicity, any product used in a food application or hand cream, cytotoxicity becomes the most important one before you prove its point. Yes, it can kill all the bacteria, but it can also kill human being also, yeah, okay. So any other question? Okay, I'll just draw one experiment here, you draw the conclusion, right. There was a school kid, yeah. Teacher kept 3 beakers. In one there was alcohol, yeah. In one it was smoke. You can take it a cigarette smoke or any smoke. In third one it was a clean air. One insect was added to each of these jars, yeah. Insect in this died, insect in smoke died, insect in this survived. So what is the conclusion of the experiment? Anyone want to conclude this experiment? Come on, this is a very simple experiment. Everybody said alcohol kills insect, smoke kills insect, clean air is good. But one little Johnny, he concluded as long as you are drinking and smoking, there won't be any insects in your stomach, yeah. So drawing the conclusion of the experiment should be done based on logic. You can draw any conclusion, yeah. A lot of such stories are there in science where we do experiment and we draw wrong conclusions, yeah. So that's why it is very important when we arrive to the conclusion, suggesting anything, to you weigh all the aspect of it, then only you propose, yeah. Because what looks straightforward may not be easier. There is another experiment related to a cockroach. Dissection was going on, somebody cut one leg, asked cockroach to walk, cockroach crawls. Then he cuts another leg, asks cockroach to walk, starts walking. Like that he cuts all 6 legs and cockroach couldn't walk. What was the conclusion? After losing all 6 legs, cockroach became deaf. That could be a conclusion, right? But insect could not walk because of leg. But the conclusion was written after losing all 6 legs, cockroach became deaf. That means he was no longer taking instructions, yeah. And you will see number of different features, because whatever experiment you are doing, I will ask you to do all the aspects. Some I have already covered, some will ask the question when you finish off your experiment. Now any question related to the experiment what you are doing? No? Whenever I ask question, everybody starts looking into their book as if they have written the piles of question, yeah? Yeah? No? So with this we conclude today's one, and you are going to go to the lab to see your experiments, yeah? So welcome back. So we'll start with what we have done previously. Before recap, who is willing to give an explanation or at least say the process what has been done? Anybody? Don't worry, if it is wrong also, you will not forget. Very good.
Sir, about the cells, the cell is first, as we all know, the composed of CaCO3, chitin and protein. First we have to demineralize with the help of the HCl, and after that we used to deproteinize with the help of protein. We will get chitin, and after we uses deacetylation with the help of NaOH, we will get chitosan.
So what you exactly did?
Sir, we means procedure, sir.
Yeah.
Sir, first we take HCl. We put HCl in the
HCl was 0.2 molar.
0.2 molar, sir.
Okay.
And after that we left it for 2-3 hours. Then we left it for drying, wash it.
Okay.
And after that we put NaOH 0.5 normality molar, sir. NaOH.
Okay. Nowadays it's only molarity which is used. Normality is no longer in existence. That's fine. Yeah. Good. Yeah.
Yes, sir. After that we left it in the oven for overnight or 6-7 hours. Yes, as sir told us.
That part is not yet done.
Yes, sir. Not yet done,
Yeah.
sir.
Okay. So basically when we try to see the process, it's better to go in a very, very systematic order. Yeah. It's good. Otherwise it becomes difficult. That's why we call it SOP. Yeah. So first was, the background was shells were collected. Yeah. You can ask all those questions to your instructor who went to different places to get the shells for you. Yeah. And it's a best saying, seeing is believing. When you see it, then you feel that it is how easy it is to do it. Yeah. So shells were dried. So one option is you grind it. Since the hard shells are there, grinding takes too much of energy to grind it, and you need a specialized type of blades to grind it. Sometimes cement, concrete crushers are used to grind those type of shells. Yeah. So these dried shells, as your colleague mentioned, you do first demineralization followed by deproteinization. Yeah. So then deacetylization in another step. Yeah. So what was the thing? You took 1:20 ratio, 0.2 molar hydrochloric acid, and the other acid was acetic acid. Yeah. That was basically 2%. Yeah. 2% acetic acid solution. So it is better to go back to your basic chemistry book, understand molarity and understand percentage of solution. Yeah. That is very, very important and very, very basic. Yeah. So in school, when we all studied science medium, nobody is from arts and humanities, right? All are from science background. Yeah. So what is 2% of acetic acid solution corresponds to how much molarity? Yeah. I use these two solutions interchangeably because if we are telling an industry, you just tell them that take 2 kg of this, mix it in 100 liters of water and use it. Yeah. If you go to a fisherman, said take 2.2 molar, what he will say? He won't understand. Yeah. And there is no point in explaining all those things. Their purpose is to solve the problem. Yeah. So there what scientist lacks. Yeah. You go to a doctor, he gives you medicine. Yeah. You don't ask how it is being encapsulated, whether the paracetamol is encapsulated into starch or how the paracetamol has been crystallized. Yeah, those are the scientific questions. Yeah, whether that starch in the tablet is of tapioca or from banana. Yeah, he doesn't explain you all those things. Yeah, because that's of no point in doing it. But when one pharmacist is talking to another pharmacist, their explanation makes sense. Yeah. So this is what you have to have clarity. Sooner you get into your head, it makes things easier. If you cannot explain a process to 6-year-old child, that means you yourself don't understand. That's the famous saying by one of the great scientists. Yeah, make sense? So molarity and percentage of solution. It's very simple. I'm not going to go into this one, but you should be able to understand that. Yeah, molarity is quite easy. You just get the molecular weight and measure it into ml. Yeah, so similar thing becomes percentage or so. Yeah, A. Second question is, why only these two acids? What if I use H2SO4? Yeah, what if I use HNO3? What if I use lemon juice which contains citric acid? Yeah, what if I use a sugarcane juice, add acetobacter aceti to make acetic acid? Yeah, if you want to try experiment, buy egg, chicken egg, put into domestic vinegar, leave it for overnight. Next day morning you will see there is no shell left, only thin layer of skin is left and all egg and albumin will be there. So this is the way shells can be dissolved. Yeah, it is not only for shells what you have done in NIT over here. Yeah, so these are the couple of questions you should be asking yourself. Yeah. Second one is ratio was 1:20. What if you take 1:10? What if you take 1:5? What if you take 1:50? Yeah. So here you have to get the best trade-off between amount of larger the solution you take, better will be the extractability. But you have to draw a line, because then you have to remove that water or you have to discard that water. Yeah. That is another condition. Then comes economic value or economics of a process to decide what is the best part. So every stage is optimized, every unit operation is optimized, and after this leave it for 2 hours, 3 hours, overnight. Yeah, and what temperature? If you add a temperature, if you add a stirring, how different it will be. Yeah. Think of simple example, you have 100 ml of water, you add 10 grams of sugar, and without a spoon it won't mix. That means stirring will help you in this particular process. If you stir the samples, whatever CO2 is dissolved, it will escape the environment. Yeah. When I say environment, that means environment of that beaker where the sample and biomass was there. So all these things comes when you observe the process. Yeah. Then only you become clear of each and every step. Yeah. Now for that I don't have to read 3 paragraphs of procedure to come up all these points. Yeah. Same thing, if whatever you do, if you make a flow chart, think over it. Why did you do it? Why? What? And how? Yeah. Why you are doing it? Why you are adding, or sorry, why, what are you doing? Yeah. Whatever you did, why did you do it? And then how? So these 3 questions at every stage will make you perfect with the process. Yeah. After this, what was left was the shells were very soft. Yeah. Because cement is gone. Yeah. Something like this, a concrete wall is having a cement, plus you have sand, metal frames and everything. That's the structure of shell. When calcium carbonate, chitin, protein, and if you want to see the structure of a shell, go into any standard book, you will be able to see that layering of that structure. Something like when you are plastering a wall, if you put a small thin metal sheet, plaster it, then put a metal rod in it, plaster it, it becomes very strong wall. Yeah, the same thing it happens. So what you are trying to do is trying to use a solvent to remove one thing which is creating the strength of the structure. So calcium carbonate, protein, this is the structure. So sequentially you are removing calcium, which is easier because a larger quantity it goes off. Then you are separating the protein, which is again a source of good quality protein, and then you get your chitin. Yeah, you went ahead for deacetylization, which you haven't done it. Yeah, but that is only to convert chitin into chitosan, and chitosan is the one which is also used for making polymeric material. Yeah. Now the challenge is how you can make the whole process in 2 hours, 3 hours, how you can do it in a continuous fashion. All those things are, this is just basic procedure. Yeah. Then you introduce a technology. So if when you, that's the reason when you read a paper, oh somebody used a microwave for production of chitin, yeah, somebody used A, B or C technology. Yeah. Now the question for you is demineralization followed by deproteinization. Can we do other way around? What we did, we first removed the calcium and then we are removing protein. What if we first remove the protein and then we remove minerals? It is possible. Yeah. So that is both interchangeable. Yeah. So that should be one of the questions, why we are doing demineralization first. We could have done deproteinization first. Why deproteinization only with alkali? Why not with protease enzyme? Yeah. That is also possible. Yeah. So the step for getting to chitin, demineralization, deproteinization, acetylation. But demineralization and deproteinization can be interchanged. Yeah. So if you are doing experiment, you will do those both the process and check the quality, check the yield, and then you come up with the conclusion. Yeah. So this is what you should be seeing in when you look into the publication. Not on downloaded 10 different papers of chitin extraction and then one step forward, 2 step backward, making situation awkward. Yeah. So then you will be only lost in the papers. So to do anything, important aspect is get started. Do some process and then try to improve that particular process. Yeah. How many of you know to make chapatis or dosa? Perfect round dosa which doesn't burn. That's very good. Did you get it in one go? It took multiple time to get a practice, right? Yeah. Same thing. So more and more you do it, same operations, then only you learn better and better operations. Yeah. So this was all to do with practical. So tomorrow what you are going to do is we are going to remove alkali portion. Yeah. And alkali portion will precipitate the protein from shells, and you will also have a fish sample which is dissolved in alkali and a water. Yeah. And then the residual part and the soluble part. The soluble part with the water or with an alkali, when you adjust a pH of 4.5, yeah, this is called isoelectric point of protein. That means protein is least soluble at this pH. But how do we determine this particular point? Why 4.5 pH? For this is also established via experiments. Very similar when you use a nitrogen as a factor of 6.25, 5.7, 6.38. How it is done? If you are extracting a fish protein, the alkali solution what you have, you make pH of 3, 4, 5, 6, 7, 8, 9, whatever, yeah, or even 2 and 1. Alkali solution you have, adjust this pH, and then wherever you see the maximum precipitation, that becomes your isoelectric point, yeah, and that differentiate, but majority of the protein will precipitate at this pH. But very same protein what you get, leftover residue will have acid soluble protein, that is called gelatin or collagen from fish, that will solubilize in acid solution, that is in and around this pH, that particular residue. So if you add acid without removing the residue, then you are again mixing everything together, yeah. So what will happen after adding alkali, you will centrifuge it, alkaline solution you will adjust a pH of 4.5, and once pH is adjusted, you decant the top solution which will still be containing some of the free amino acid, some of the nutrients, acidified water, that water is a waste water, whereas that can be used easily for production of duckweed, macroalgae or microalgae. That's only you will be able to introduce circularity. Now you might have a question, if you have to use such a small amount of water, but this is only experiment, you are doing a smaller quantity. But if you are dealing with 50 grams, you might get in the best way possible from crabs around 5 to 6 grams of chitosan. Yeah? Okay. If this is 50 gram, to get 5 to 6 grams of chitosan, you are already using 2 liters of water. Yeah? Think to make 100 kg of chitosan, how much shells will be required and how much water will be used. Yeah? Now this is the volume what we are talking about. Yeah? So that's the way how things goes up in the order. Yeah? Now by doing so, you are able to get all your chitin or chitosan from shells. Yeah? Any questions so far related to experiment? Now going back to production of duckweed. How do we produce duckweed? Specimen putting in a water and it just multiplies. Yeah? Something like when you want to spread a bacteria, how you spread a coronavirus, exactly the same format. Yeah? Macroalgae. Macroalgae can be grown directly into from spores. What you saw the commercial production, thin rope is tied into one particular metal rod, dipped into the spore solution, taken it out, rope it around a bigger rope, throw it into the sea, forget it. Yeah? That's it. No fertilizer required, nothing else to be added in. Nature will take care of its own and it will produce huge amount of biomass. So per hectare you have a different type of yield you can obtain. And some of the seaweeds can also be cultivated on land with normal pH water. Yeah? That is neutral pH. So when we grow seaweed in ocean or sea, it needs saline environment. What is sea salinity? Loud.
Salt content in ocean.
How much is salt in the sea water? You got Mike by surprise, isn't it? It is 3.3 or 3.4%. Yeah. Have you heard of a sea called Dead Sea? Is there in near about Turkey. Their salt is so high, you can even swim or you can walk on that particular sea. Yeah. In ocean, sea salinity is 3.2 to 3.5% or 3.3%. That much amount of salt is present in it. Yeah. And this is the condition which is required for marine algae to grow. And this much amount of salt water is very high, and for food preservation you need only 0.5 to 1% maximum, making a pickling another one. So other one is how best you can use the sea water for making your own pickle. Yeah, getting the sea water, boiling it and then using it for making pickles. Yeah, then you sell it as no added salt. Technically you are correct, practically you are correct, technically you are wrong. Yeah, because salt was already there. Yeah. So we covered duckweed production, macroalgae, microalgae. Yeah. I have one of the bad habit, when I see something, I'll go and see how it is being produced. If you want to cook a best sweet, go and observe when somebody is cooking it. You did not learn by taking a lecture. You learned how to make dosa or chapati by seeing how your mother is doing it. Yeah. There was your interest, either by force you learned it or based on your own interest. Yeah. So when you observe it, ask yourself questions and then you will never forget. Yeah. So that's was what we have covered so far, and now we go on to how do we establish biorefineries? So there will be 3 sequences. One is biorefinery for fish, biorefinery for shells, and biorefinery for aquatic plant. So some of the stuff will be repeating what we have already spoken about. So first and foremost question is, what are biorefineries or new type of biorefinery? We all have studied petroleum biorefinery, yeah? If you see, we do not call it refining, we call it petroleum biorefinery, yeah? So that is not a new word. Even we call it fossil fuel, but they are of biological origin. So once you get mining or you get a crude oil, does anybody know about the petroleum biorefinery? Once you get a crude oil, how we get petrol, diesel, wax, paraffin wax, kerosene, everything in same refinery?
Distillation process, fractional distillation.
Okay, you have a crude oil which is basically a black in color, yeah, and then you heat that oil, right?
Yes, sir.
So whatever vapors goes on top is your petrol, yeah. Then
Diesel,
diesel,
kerosene,
kerosene, then wax, paraffin wax, and all those elements, whatever. So 1 liter of oil will give you different fractions, right? That means same biomass can be fractionated into number of different compounds. Can you suggest any other compound? Yeah. So very same oil, we get number of different compounds. Whereas when it comes to food, if you have wheat grain, yeah, we have done enough for dairy example. Now let's move on to wheat. Wheat is milled, yeah, or wheat is harvested. So what we get? Outer coating, shells. Shell in the means husk, yeah, and you have a straw. When wheat is harvested, you put it into a thresher, or if you take rice, how many of you seen wheat processing? That's good, at least one has seen it. So wheat is harvested, it goes into a thresher, or if it goes in a combine, you get husk, you get grains, yeah, and then these grains goes for number of different processes. One is goes for what you call chakki atta, that is whole wheat atta used for making chapatis, or else this wheat goes through number of different processes where you get wheat germ, yeah. What else? Maida or semolina. What is the difference between maida and semolina? Maida and semolina is the same thing, different particle size. If semolina is grounded into fine flour, it becomes your maida, yeah. And then you have bran, yeah. Same thing applies for rice. This goes for different types of product. Germ mainly contains certain bioactives as well as wheat germ oil, yeah, and similar thing happens with bran as well, and the bran also contains certain type of oil plus fiber called ariboxylans, yeah. Have you heard of this word ariboxylan? Have you heard called glucans, beta glucans, yeah. Beta glucan is very well advertised and it comes from oats, that is oats fiber, soluble fiber. Same way wheat also contains ariboxylan and same oats also contain. So our main focus is wheat to this atta or this maida atta, yeah. We don't think about what we do with husk, we don't think what we do with straw. Some farmers who are much more advanced, they store this straw for feed purpose, yeah. This is mixed with other green grass to make feed for cattle. Those who have seen the villages, how this type of straw, the straw means the chopped straw mixed with the water, what do you call that word? Sani? Yeah?
Bhosa. Bhusa.
And when you mix it with the green, what you call barseem and all those things, any of those green grass or other one, and feed it to animal, right?
Normally we call it chara.
Chara, yeah. So that means this is already used as a feed, yeah. What do you call that? It will be different word in different places, yeah. Whereas rice straw is not normally used as an animal feed because it contains large amount of silica, yeah. So you would have seen even when we remove the outer polishing of rice and all those side stream which goes as a waste, to get rice germ oil or rice bran oil which is also sold in a market. So if you talk everything together, that is basically a biorefinery, yeah. So how a biorefinery looks like this. So from where the wheat will go and from where it will come. This is basically a photograph of a petroleum biorefinery, yeah, or any oil biorefinery or any of those one. If you want to see some of the coal refining unit and all, you can go in and around, you can see the steel refining here in Rourkela steel plant. This is the overall. So we try to establish biorefinery for seafood, for fish or any of the biomass. Concept remains the same, yeah. Simple one is to fractionate it. So there are number of different definitions for biorefinery. One, a facility that integrate biomass, conversion process and equipment to produce fuel, power and chemicals, yeah. So how you make an assembly together so that you can get energy, power and chemicals. This was one of the definition. Food was out of it. Very few literature you will see on food biorefineries and all, yeah. We are just in a process of completing a book on food biorefineries, and it should be coming out in 1 or 2 or 3 or 4 months, yeah, next few months. So that is exclusively on food biorefineries. It is analogous to today's petroleum refineries, yeah, or sustainable processing of biomass into spectrum of bio-based product. Food is added in, feed is added in, ingredients and chemicals and bioenergy, yeah. The circular part of the seaweed, when we are showing the biomass, conversion, fractionation and storage or whatever you want to use it as. So all these three things together will make a full biorefinery. So why we are moving towards food biorefineries? One, complying with net zero target, that means zero waste approach. To establish zero waste, that means you want to utilize every fraction of it. That means if wheat is produced or fish is harvested from the sea or seaweed is processed, every fraction of that is utilized, yeah, whether for food purpose or a feed purpose, ingredients and chemicals. So wheat was a perfect example where nothing is wasted, yeah. The straw stored, mixed with the green grass and all to make feed for animal, or you call it simple as chara for animal to eat, and same one it produces milk, so it produces another chain, and cow dung and all if you want to use it for a fertilizer, and the whole wheat grain goes into chakki atta, that whole wheat atta you make chapatis. So nothing is wasted, yeah. But similar pattern is not adopted for seafood, yeah. Majority of the work, majority of the literature, zero waste approach for dairy is well established. Dairy processing is well established, even though it's a liquid product, yeah. Every fraction of milk is utilized, whereas certain biomass is still having a huge scope to make a zero waste biorefineries. Whether you are working on insects or whether you are working on pulses, even pulses certain parts are, but not all biorefineries are well established. Second, reducing the greenhouse gas emissions. That means how we can reduce greenhouse gas emissions by valorizing it, reducing the food wastage, and you have already seen what is the impact of waste produced globally and how much greenhouse gas emission it is causing, and it can be rated as third country in terms of greenhouse gas emissions according to FAO estimates. Third, to achieve sustainable developmental goals, there are 17 such goals, directly or indirectly how you can achieve those. Transiting economies to an environment economic and socially sustainable. That means moving E to CE, yeah, or you can call it a general economy to circular economy, yeah, or general economy to bioeconomy. When you establish a bioeconomy, you can again introduce circular. So, circular bioeconomy, it becomes B, blue, when you are dealing with marine sources or freshwater species, yeah. Any questions so far with biorefinery? Now, how can we establish a seafood biorefinery? First, what is the requirement of establishing any biorefinery? We need a raw material, yeah. Determine its composition, what all it contains, that is very important, yeah. Without knowing what it contains, how you are going to establish a biorefinery, yeah. And then determine its physico-chemical properties, that means you estimate its properties, how it behaves, how you can store it, how you can stabilize it, yeah. First and foremost, to know what you are dealing with, biomass. Second, define target bioproducts and its physico-chemical properties required. Think of the shells or a fish. If you have a fish, what all it contains, composition, moisture, 70 to 75% moisture, yeah, 10 to 14% protein, any general fish, 3 to 4% oil, 1 to 2% or 2 to 4% ash, yeah, or within this protein how much is collagen. So, you determine all those composition and then try to see what all you want to get out of it. You want to get a whole product, yeah, or you want to establish seafood side stream biorefineries, yeah. Then you make an inventory of technologies or any intervention required to produce targeted bioproduct. This is very simple, plain English to establish a biorefinery. If you want to add any terms or anything is missing, you can interject in between, yeah. First to know the raw material, then what you want to obtain out of it and what are the physico-chemical properties. That means your target is to get a petrol. Petrol is having a specific density, a specific composition, its flash point, its knocking value, and all those properties are there. And based on that, the processes are established. Same way, if you are establishing a seafood or fish biorefinery, then you need to know what should be the composition of collagen protein or protein of 75%, protein isolate, protein hydrosolate, or protein concentrate, yeah. And then target bioproduct. Make an inventory of input material, yeah, that means what all is required inside. Data collect for each unit operation, that means whatever is operated you get all the unit operation, and determine chemical compounds to be transformed via pre-treatment or different processes, whether you have to pre-treat it for stabilization of biomass or for production of different ingredient. Now, once you have done all this, you have to do cost benefit analysis. LCA stands for life cycle assessment. What is LCCA? Life cycle cost analysis. Another one is TEA, techno-economic assessment, yeah. That means whatever process you are making, you make an assessment of it. So, experiment what you are doing, some of those experiments are already done, are there in the literature. You make all those questions, establish full biorefinery, then go for execution. Yeah. Before doing all these things, the first question is asked, what will be the cost of this technology? Yeah. That's a wrong question. It's the right question, but at a wrong time. Yeah. What will be the cost of phone? 20 years ago, only elite people were having smartphones, not smartphones, mobile phones, that too this big. Yeah. But now everybody is having smartphones. So, if that time the inventors of mobile phone were thought it's very expensive, we won't be here. Yeah. First you make a process, then improve the process and reduce the cost. That's the way it is always growing process. Yeah. So, the first inventor of the phone was Graham Bell. Yeah. It's not Steve Jobs who made iPhone, but the concept was there, and these are all developmental work or innovations. That was invention, and now all these things are innovations. Yeah. That's one is fundamental invention and innovation. That's the difference between invention and innovation. Yeah. Any question for biorefinery? If you want to see all these steps for establishing a biorefinery, there are clear-cut guidelines to establish a petroleum refinery or oil refineries in IEA, International Energy Association guidelines are there. How do you establish all those refineries? Yeah. So, similar type of model can be adopted for establishing any given biorefinery. Instead of C, you can have any other product also. Yeah. Or establishing micro-algae biorefinery, macro-algae biorefinery, and you will see some of those examples in coming days. So, what is the processing of biomass for valorization? How do we process it? Any sequential order? What will be the next step? First and foremost is stabilization of that biomass. If you have fish waste or a shell waste, how you stabilize it, or what type of pre-treatment you have to do it. Yeah. What was done in the case of the shells what you are using? It was cleaned and dried. Yeah. So, drying is also a pre-treatment. Yeah. When I say pre-treatment, that does not mean, oh, something complicated technology. So, conventional technique, drying in hot air, 4 to 6 hours or 8 hours. Using microwave for drying, 20 minutes, drying completed. Yeah. Or in some cases, you do not have to do any pre-treatment. So, you can use a fresh shells directly. In that case, you do not have to use huge amount of water. Now, shells which were recovered, they might be having around 40 to 70% moisture. Shells were dried to 5 to 7% or up to 10%. Yeah. Then you are adding 1:20 water. If you use directly fresh shells, which will be difficult to crush, then you do not have to use 1:20. Here what you are doing, you are removing the water, then you are adding the water. Correct? To make a coconut chutney, you can use a fresh coconut. I know nobody has tried from the dried coconut, but I have tried with the dried one as well. Still you can make it, but it takes longer and you have to add more water to it. Yeah. Using milk directly for making a tea or using a milk powder. Yeah. Same situation. So, you can use without a pre-treatment directly a fresh one. Why you are doing stabilization of pre-treatment? Any question? Or why you are doing it? I am asking the question. Why do we do stabilization of pre-treatment? Obviously, to stabilize the biomass or to improve subsequent processes. So definition of pre-treatment can be any treatment given prior to process to improve extraction efficiency, preservation of biomass, or to improve extraction yield, number of different rules. Now giving an example, how many of you know about blanching? Who is going to define it? Okay, give it, yeah.
Dipping the fruits or vegetables in hot water for easy removal of the peel, potato.
Sudtanni.
Sudutani.
Correct. What temperature?
Hot water. Hot water temperature.
Okay. That you said about hot water. Okay. Hot is, so what is hot for you? 40, 50, 60? I'm asking water, so you can answer it.
I don't know, sir. Yeah.
Okay. And who was the other one for blanching? Why I'm asking you this? Because this is most commonly studied process in food, right? Yeah.
So blanching is done for removing extra foreign substances also. There are two types of blanching. One is hot water blanching and steam blanching. So according to our needs, the selection of blanching will be done.
Okay. In this case, hot water will be what?
80 to 90 degrees Celsius, I guess.
Okay. And what's the purpose of blanching?
Enzyme inactivation for plants, fruits and vegetables.
That's fine. Enzyme inactivation. Any other?
Sir, it delays ripening and
Blanching. When you put a fruit into hot water, it will not ripen, it will cook. No? No, this side, okay, go on, yeah. And any other step why blanching is done?
So blanching is a pre-treatment operation to inactivate the enzyme in fruits, especially polyphenol oxidase.
That's enzyme inactivation, yeah. Any other thing why we blanch it?
Sir, it retains the color retention.
It retains the color, okay. Any other?
It loosens the cellular structures of the fruits and vegetables.
Okay. So these all are very, very technical. How many of you have seen, how many of you made a palak paneer or other one? You've seen spinach is a big volume, yeah? You add into the hot water. So are you inactivating enzyme there? No, right? One is stabilizing the color. What blanching does? Your friend mentioned about your what you call stopping ripening. Sometimes mango, fruits and all, they are mixing of 2 or 3 concepts were together, yeah. Blanching is simple as what your friend clearly defined as dipping in hot water or steam. This is done for multiple purposes depending upon the product. If you take a potato, if you slice it, howsoever your mother might be making potato chips, yeah, they dip it in hot water. If you don't dip in hot water, if you fry it, it becomes brown because of polyphenol oxidase, yeah. How this enzyme get activated? Potato is there, it doesn't turn brown. Apple doesn't turn brown. When you slice it, it turns brown, yeah, because oxygen it gets from the air. Enzyme is present, phenolic compounds are there, it acts and makes quinones, yeah. So the moment you dip it in hot water, this enzyme loses its activity. So you don't have to blanch it to inactivate enzyme. 2 ways: cut the oxygen supply, yeah, or make enzyme not to be active. If you slice an apple at 2 degrees Celsius, it will not turn brown, yeah. So you don't have to use energy for that purpose, yeah. So any of the unit operations, that's why you have multiple definition of blanching, because if you would have read the paper, if you read the book or different chapters, everywhere explanation is different, yeah. Nothing is wrong, everything is correct, but you have to see what was the intention of it, yeah. Second one, that was for potato. Other one, hot water dipping is also done to kill certain insects like cauliflowers, yeah. You dip in hot water before cooking. Certain vegetables are dipped in hot water. Cabbage, the moment you dip in the water, it squeezes, volume is reduced. So to remove occluded oxygen, yeah. This explains why color is retained in blanched sample. Oxygen is there, it will oxidize the color, yeah, correct. So, for that we remove oxygen trapped in the air into the plant biomass. The moment you blanch it, air is gone, then it settles down, yeah. So, there are multiple one. So, this is also one of the important unit operation which is done in a processing environment as well as in stabilization of biomass, yeah. Do you agree? The reason I used blanching word, because this is one of the most common unit operation we all study, yeah. Whether we are food or non-food people, we do study blanching, yeah. Blanching before chips, blanching making before spinach curry or any of those food and food ingredient, yeah. Next step goes extraction. By using conventional extraction. How many of you used maceration before? Have seen the maceration, yeah? Those even we macerate it in ginger and different crops or different biomass. That is one of those conventional extraction, or also known as solid liquid extraction. What you did with the shell was simple solid liquid extraction. Other one is by using new technique or hybrid. Hybrid, that means mixing both conventional and new, using ultrasound with the solvent, yeah. That will be ultrasound assisted or microwave assisted, or in some cases you do treatment direct to extraction or direct extraction, yeah. So, when you look into the literature, it will, some studies will show ultrasound as a pre-treatment for extraction of ABCD, yeah. Some places it will say ultrasound assisted extraction. So, remember, there is no clear cut definition between or no boundaries between these two words, yeah. Okay. Then we get end product. So, this is the step for processing of any given biomass. Now, come to fish, yeah. So, these are some finer details. I have one of our PhD from Inner Mongolia in China. She is working on establishing full biorefineries for seafood side streams, yeah. So, these are the waste what we get from fish alone, yeah. NaOH, what you are going to do, yeah, homogenize it, 0.1, 0.2 molar different concentration of acids, yeah, and this is pretty much very standard process, yeah. And then isoelectric solubilization, that means you solubilize the protein, you centrifuge it, this is all what you will be doing, and then you can get lipids, insoluble bones and skins, or else if you are using an enzyme, proteases, you will get your peptides, yeah, or protein hydroslate. If you take a fish side stream, you mince it, what you will get is a fish paste. If you add alkali, protein will solubilize, you precipitate, when you centrifuge it, you get multiple layers, yeah. Then you can separate oil, protein and bones and all, yeah. Very similar, whatever you have done with the shells, same thing can be done over here. And how and where and what should be the step followed, and whatever is left over insoluble fraction, if we do acid extraction, we can get collagen, yeah. It is pretty standard process. You go, even if you put it on normal browser, fish protein extraction, you will see the steps. Put fish oil extraction, get that process. Fish protein extraction, you get that flow chart. Fish collagen extraction, you get that flow chart. Put all those three flow chart together, identify the common step and draw your own process line to obtain multiple ingredients out of it, yeah. That is the best approach. Oh, sorry, this one. Now, if you will see fish biorefinery, target was to produce protein and peptides. Yeah. Minced fish, this is what experiment you are doing, adding into a water, or you have added already, yeah, in water or in alkali. You are not doing this part. After this, when you centrifuge it, you get your supernatant and you get your residue. Yeah. This supernatant, if you precipitate with acid, you get your fish protein. How many of you disagree? As I already mentioned, there may be a lot of mistake in this. You have to pick those mistakes. Yeah. Otherwise, you will be learning wrong things. And I won't be responsible for that, right? So, you get a protein. No, nothing is wrong in this. Yeah. So, there is no point in scratching your head in this case. So, you get your protein, and whatever is left to a residue still is having insoluble proteins, that I said already, collagen or gelatin. Strategies are either to use enzyme to break those protein into peptides or protein hydrosolate, or else use acid for extraction to get collagen. Yeah. And then this is your product B. Whatever A, B, C, D is written, it is clearly written here what does purity and what are the purities it is having. Green color is product A, blue is product B, and C is product C. Product A alone, 1, 2, 3, 4, 5, 6, these are the treatment. If you use only water, because majority of the fish protein is soluble in alkali, yeah, and I am not going to go with the ultrasonics and all unless and until we know about those technology. That will be lecture 16, 17 and in coming days. Control number 4 in alkali, and you can see how much you can get out of it. Yeah. So, that means protein is of fish is alkali soluble. Here what was used was 0.1 molar, what you are using is 0.2 molar. Now, the question is, if you use 0.5 2 molar alkali, will you get more protein? This is also very important. One is the pH. Yeah, we can achieve pH 12 by using alkali, yeah, or take acidic one. You can achieve pH 3 by using hydrochloric acid, H2SO4, citric acid, acetic acid, lactic acid. Then why we mainly focus on HCl or NaOH? Why not H2SO4? Why not nitric acid? Simple answer is it is easy to handle. Yeah, and this is mineral acid. It gives H plus. Yeah, H2SO4 will be better option because it gives 2 hydrogens. Yeah, sorry, 2 hydrogens. So, what is the relationship between pH and hydrogen ion concentration? Question to you guys is, if we use higher molarity solution, will you have a better extractability or no? Yeah, because there is no relationship between acidity and a pH. Yeah, though it looks like that, because pH is negative of hydrogen ion concentration, varies from 1 to 14. Yeah, but if you have a larger molarity of solution, you may not be able to solubilize the protein, because when there is H plus, you need OH also. Yeah, and that is why when you adjust a pH, it is not that if you use 5 molar hydrochloric acid, you need only 1 or 2 drops. That is wrong conception. Always you take a weaker acid to precipitate it. Yeah, think of when you have to make cheese, when you have a lemon, you dilute a lemon, then you add into it, then there is a phase separation proper. Why, if you use directly a powdered citric acid into milk, it will not curdle properly. Yeah, have you seen it? You make a citric acid or tartaric acid, you make a solution, then you add it slowly while churning it around. Yeah, have you ever asked why it is done? I am not talking about the professors of dairy chemistry or dairy processing. Ask the person who make lot of cheese, chhena and all those sweets. He will never add lemon directly. They will add lemon into the water, mix it, then they add it. Tartaric acid or citric acid, they add into a water and then they add slowly while stirring it. Yeah. So that is learning by experience. Same thing happens here. Yeah, because the same protein, not same protein, basically protein made up of same amino acids. Yeah. So as you can see, this is having this product A as these two values, and then the residue, if we use enzyme, yeah, there is specific procedure for that, and you are getting this much amount of protein recovered, both product A plus B, that is, this is also protein and this is protein hydrolyzed protein or protein hydroslate, and you can see how the recovery is changing. Yeah. When it comes to the purity, that means how pure is the protein, green one is product A here, with water you see 30, 68, 66, 75, 73. Yeah. So you optimize a specific condition to come to a level, what should be the alkali concentration, how much time you have to use it, what technique to use it to get the protein. The next task comes, what you are going to recover next. Yeah. Then when you are left over with bones and scales. Yeah. The similar operations with different techniques. HPP is a high pressure. Yeah. So not to worry about the technology, we will cover that one. Yeah. Here the ratio was only 1:4, not 1:10, because fish used here was fresh fish, minced with lower water, and then we use two different techniques, one after the other, and now your recovery of these ones is improved. Yeah. So you use a technology based on what is your intention, 82% pure, and recovery is increased in every case. Only with water alone you have higher recovery. Yeah. Yeah. So that means high pressure was able to solubilize more of protein into water. Yeah. So these are some of the experimental data, not to worry about all these numbers and a treatment, that all you can play around with yourself. Yeah. So this is very simple process. Bottom line is you have biomass. Here you can call it high pressure as a pre-treatment. Why high pressure was done? It's not because technology is there, that's why it was used. High pressure was done mainly to inactivate microorganisms. Yeah. When you have a fish biomass, this fish came from the factory, side stream, it was left out, so there will be some microbial contamination, and that microbial contamination will come into this protein because we are not heating anywhere. Yeah. So we gave a high pressure treatment to inactivate microorganisms rather than heating it, but by giving high pressure treatment we got a microbial inactivation but increase in recovery yield. Yeah. So that becomes the interesting part of the data. Yeah. So here precipitation was done 5.5. This was, again you will investigate what is the isoelectric point. Every fish, source of the fish will have different precipitation point, and this was also changed because of this pre-treatment, because high pressure can also alter the properties of protein. Yeah. So that is one simple example for protein purity and followed by enzymatic hydrolysis. Yeah. Now other one is for oil. Oh, sorry. Other one for oil, fish drying. I don't do all this animation, this is what keeps happening. Yeah. So for this, again either you have I'll leave it on to this one. either you have a supercritical condition or direct oil extraction. Yeah. So that means supercritical, that means you are using liquid CO2 to obtain proteins and followed by alkali treatment to get your protein and lipids together. Yeah. So do you have any way how you can extract your, I'll just leave it on here, extract your oil, protein and peptides? How we can extract oil, protein and peptides together from a biomass? So in the case of fish, it is possible to do drying of fish, yeah, get your oil out using supercritical, yeah, and all the carotenoids and pigment leftover biomass, then to go for protein. Or else if you have a fish which contains 14% moisture, sorry 14% protein, 70% moisture and 5% lipid, the moment you dry this fish, 5% lipids might become 20 to 25 because you are eliminating the moisture, 14% protein may become 45 to 50%, correct. Then if this goes for supercritical fluid extraction or hexane for recovery of fat or lipid, then again this protein may become from 50 to 70%. This is complete dry fractionation route, yeah. And bones and scales can be removed by using simple sieving technique. So when it comes to extraction, either it's a wet fractionation or dry fractionation, yeah. Now any questions? Supercritical or direct to remove oil, that's lipid, and then followed by alkali treatment or pressure treatment, or to use water to get your proteins, yeah. Any questions? That was a good bit of entertainment, swinging in and out, yeah. Any questions or any question related to protein, lipids or peptides, or how do we establish biorefinery? No? Or any suggestion? So this is really important to see how we can process the biomass, and there are number of techniques, number of conventional ways we can obtain our biomass. So one, establishing a biorefinery, you need to know about the biomass. How do you stabilize so that you can store it for longer? Develop the extraction processes, optimize each and every condition, and then check the quality of the product. If it is not as per your specification, then you alter your extraction technique or condition, or you alter your pre-treatment strategy, yeah. So quick recap is about how do we establish biorefinery. One, you need to know about your raw material, its composition, A, most important element, and understand its properties. Second, what are the target bioproduct? What all we can get from fish is your protein, lipids, yeah, gelatin or collagen, minerals, yeah. And once you have a lipid, then you can get easily your glycerol and fatty acid for further fractionation, yeah. So this is all how you can establish a complete biorefinery by establishing different unit operations, yeah. No questions? No. So tomorrow what you are going to do is you already have alkali solubilized with fish protein, and you have shells protein. You will precipitate that with 4.5 or 5 pH. You will see something is precipitated at the bottom. Once you centrifuge it, bottom one will be protein. If you measure it, it will be around 60 to 70% protein in it and the liquid portion. Once protein is done, this is in the case of fish, whereas in the case of shells, when protein is separated, leftover biomass is all chitin, and that can be converted into chitosan by using high amount of alkali, 50% at higher temperature. So, it depends upon your instructor if they want to convert that chitin into chitosan, and you can check its property with commercially available chitosan. At least you can tell, at least this much you can tell, was that experiment difficult or easy? Yeah? Whole of research itself is easy, but we need to be sure of what we want to do and what we want to extract. If you don't know what you are doing, it becomes difficult. Remember, this world is full of two types of researcher. One, they manage what they understand. Second, they manage what they don't understand. Second category is the people who want to manage to show that they are doing complicated stuff. Yeah, that is where the most of the problem lies. Yeah. So enjoy your enjoy your rest of the evening. Yeah.